This leaves next to nothing for the polar bodies that follow, which in most species die without function, while in some species they take on different special functions. In tissues, including the liver and skeletal muscle, another form of mitosis occurs without cytokinesis, resulting in multinucleate cells. Under the plasma membrane and constricts the cell into two halves. By fusing intracellular vesicles, a new membrane is incorporated into the plasma membrane adjacent to the contractile ring at the same time. This extra membrane is needed to cover the additional surface area caused by cytoplasmic separation. As a result, there are four steps of cytokinesis: initiation, contraction, membrane insertion, and completion. Figure 2.95 Cleavage of an animal cell SEM. The appearance of a cleavage furrow, a shallow groove in the cell surface near the old metaphase plate, is the first symptom of cleavage. A contractile ring of actin filaments surrounds the furrow on one side connected with myosin-like protein molecules. The myosin molecules interact with actin microfilaments. The ring contracts as a result of this. The expansion of the separating cell's microfilament ring is equivalent to stretching a drawstring. The cleavage furrow deepens until the parent cell is split in half, resulting in two entirely different cells, each with its own nucleus, cytosol, organelles, and other subcellular structures. Due to the extreme rigidity of plant cell walls, plant cytokinesis varies from animal cytokinesis. Instead of a cleavage furrow developing between plant daughter cells, as it is between animal daughter cells, a cell plate develops in the cytoplasm and expands into a new, double cell wall between plant daughter cells. The cell is, thus, divided into two daughter cells. A specialized type of secretion is found in higher plant species known as cytokinesis. Golgi-derived secretory vesicles containing cell wall materials are transferred to the equator of dividing cells at the end of anaphase. The cell or spindle midzone fiber. The central spindle is needed for effective cytokinesis in a variety of organisms, including H. sapiens, D. melanogaster, and C. elegans, but the phenotype synonymous with its exclusion differs from one species to the next. For example, some Drosophila cell types are unlikely to form a cleavage furrow without the central spindle, while a cleavage furrow is observed to shape, ingress, and then regress until cytokinesis is complete in both C. elegans embryos and human tissue culture cells. The drop in CDK1 activity during anaphase induces meiotic spindle reorganization and central spindle development. At the metaphase anaphase transition, CDK1 activity decreases, resulting in dephosphorylation of inhibitory sites on various central spindle components. To begin with, the extraction of the CDK1 phosphorylation from a subunit of the CPC chromosomal passenger complex enables it to be translocated from the centromeres to the central spindle during metaphase. CPC plays a role in the phosphorylation of other central spindle elements, such as PRC1 microtubule bundling protein needed for cytokinesis 1 and MKLP1, a kinesin motor protein. PRC1 forms a homodimer that selectively attaches to the interface between antiparallel microtubules, enabling spatial organization of the central spindle's microtubules. PRC1 was previously inhibited by CDK1 mediated phosphorylation. The central spindling complex is formed by MKLP1 and the Rho family GTPase activating protein CYK4, also known as MGCRAP. In brief, the self-assembly of the central spindle is triggered by the phosphorylation of several central spindle components caused by the decrease of CDK1 activity at the metaphase anaphase transition, either explicitly or implicitly. The regulation of cleavage furrow placement, the transfer of membrane vesicles to the cleavage furrow, and the development of the mid-body structure needed for the final phases of division are all possible roles of the central spindle in cytokinesis. Plate, which is formed by the fusion of these vesicles, is a membrane-bound compartment. The cell plate extends centrifugally from the center towards the periphery until it enters the zone of attachment. Following this binding, the cell plate goes through a complex maturation mechanism wherein the sugar molecule of callose is substituted by cellulose and pectin. Cytokinesis is similar to binary fission in prokaryotes, but the mechanism varies in prokaryotic and eukaryotic cell architectures and functions. In contrast with the linear chromosomes of eukaryotes, a bacterial cell has only one chromosome in the shape of a closed loop. As a result, bacteria do not form a meiotic spindle during cell division. Often, in prokaryotes, DNA replication happens after chromosome separation. In mitosis, duplication occurs during the interphase, although the daughter chromatids do not fully differentiate before the anaphase. The main challenge for a cell going through cytokinesis is to make sure it happens at the correct time and in the correct place. Cytokinesis must not start too early in M phase, or the course of the dividing chromosomes would be disrupted. It must therefore take place at the appropriate time to correctly divide the two separating sets of chromosomes so that each daughter cell receives a complete set. Cytokinesis in animal cells. During the anaphase of mitosis, animal cells initiate cytokinesis shortly after sister chromatid separation. Anaphase spindle reorganization, division plane specification, actin myosin ring assembly and contraction, and abscission are the distinct steps in this procedure. The strict temporal integration of the above individual events by molecular signaling pathways ensures accurate genome partitioning to emerging daughter cells. 1. Anaphase spindle reorganization, the stabilization of microtubules and reorganization of the meiotic spindle to form the central spindle are the first steps in animal cell cytokinesis. As non-kinetochore microtubule fibers are wrapped between the spindle ends, it forms a central spindle tree. Actin myosin ring assembly and contraction. The actin myosin contractile ring induces the cleavage process at the cytokinesis furrow, through which the cell membrane and wall expand inward, pinching the mother cell into half. The filamentous protein actin and the motor protein myosin 2 are the two main components of this ring. At the cell cortex adjacent to the cell membrane, the contractile ring assembles equatorially in the center of the cell. In animal cells, the Rho protein family, Rho protein in mammalian cells, is an essential regulator of contractile ring formation and contraction. Two key effectors of the Rho pathway facilitate the assembly of the actin myosin ring. Rho triggers diaphanous-related formins, which promote the nucleation of unbranched actin filaments. The development of contractile rings depends on the local generation of new actin filaments. Propylin, a protein that attaches to actin monomers and helps load them onto the filament end, is also required for actin filament formation. Rho also facilitates myosin 2 activation by the kinase ROCK, which phosphorylates the myosin light chain to activate myosin 2 and inhibits myosin phosphatase by phosphorylating the phosphatase targeting subunit MYPT. The contractile ring also includes the scaffolding protein aniline, in addition to actin and myosin 2. Aniline binds to actin, myosin, ROA, and CYK4, binding the equatorial cortex to the central spindle signals. It also assists in the actin myosin ring's connection to the plasma membrane. Another enzyme, septin, has been suggested as a structural scaffold for the structure of the cytokinesis apparatus. Contraction of the actin myosin ring causes ingression of the attached plasma membrane, which divides the cytoplasm into two regions of developing sister cells after it has been assembled. The motor protein myosin 2 generates the force for contractile processes by moving around actin. Myosin 2 moves through these actin filaments, constricting the cell membrane and forming a cleavage furrow, using the free energy produced as ATP is hydrolyzed. 2. Division plane specification. The development of cytokinetic furrows and the determination of division planes are the second steps in animal cell cytokinesis. To avoid chromosome destruction, the division plane between the two masses of separated chromosomes must be precisely positioned. Additionally, the process by which the spindle decides the division plane in animal cells is one of cytokinesis, most persistent mysteries and a source of heated debate. Furrow induction can be explained in three ways. The astral stimulation theory proposes that astral microtubules from the spindle poles deliver a furrow-inducing stimulus to the cell cortex, where impulses from two poles are somehow directed into a loop at the spindle. A second theory is that the cleavage furrow is caused by a constructed signal that stems in the central spindle equator, which is known as the central spindle hypothesis. By fostering the concentration and activation of the small GTPase rho at the equatorial cortex, the central spindle may aid in the specification of the division plane. The astral relief theory is a third possibility. It is hypothesized that active actin myosin bundles are spread across the cell cortex, and that inhibiting their contraction at the spindle poles results in a gradient of contractile activity with a maximum activity at the midpoint between the poles. To put it another way, astral microtubules send out a negative signal that causes cortical relaxation near the poles to increase. The spindle sends two superfluous signals to the cell cortex, one from the central spindle and the other from the spindle aster, according to genetic and laser micromanipulation experiments in C. elegans embryos, indicating the role of various pathways in the positioning of the cleavage furrow. The relative value of various signals differs between cell types and species. Additionally, the system's robustness and spatial accuracy can necessitate a large number of signals with partial redundancy. 4. Recycling of waste membrane and other materials from a cell plate, and 5. Fusion with the parental cell wall. Figure 2.96 Cell plate formation in plant cell chem. Plant cells, which have cell membranes, have distinct cytokinesis. There isn't a cleavage furrow to be seen. During telophase, vesicles from the Golgi apparatus travel through microtubules to the cell center, where they coalesce to form a cell plate. If the cell plate expands, the cell wall materials carried in the vesicles accumulate within the cell plate. The cell plate expands to the point that its surrounding membrane fuses with the plasma membrane along the cell's periphery. The outcome is two daughter cells, each of which has its own plasma membrane. In the meanwhile, between the daughter cells, a new cell wall has formed from the contents of the cell plate. The locations of the two daughter cells in relation to adjacent cells are determined by the orientation of the cell plate. As a result, modifying the planes of cell division, as well as enlarging cells by expansion or growth, results in various cell and tissue forms, which help define the plant structure. The phragmoplast is made up of the meiotic spindle remnants. This cleavage furrow ingresses moves inwards as a result of continued hydrolysis, a striking mechanism evident through light microscopy. 4. Abscission. The actin myosin ring reaches a diameter of about 1 - 2 micrometers when the cytokinetic furrow has ingressed, until a mid-body structure composed of electron-dense, proteinaceous material is formed. Most animal cell types remain bound by an intracellular cytokinetic bridge for many hours before abscission, the final stage of cytokinesis, which is an actin-independent operation. Abscission is a structural mechanism that splits the mid-body into two halves. The elimination of cytoskeletal materials from the cytokinetic bridge, constriction of the cell cortex, and fission of the plasma membrane are all steps in the abscission process. The central spindle produces thick bundles of antiparallel microtubules, which are located in the intercellular bridge. Spastin, a microtubule severing enzyme, is primarily responsible for microtubule bundle disassembly within the intercellular bridge. The underlying cytoskeletal components must also be extracted for complete cortical constriction. Cytokinesis in plant cells. Plant cell cytokinesis differs greatly from animal cells due to the presence of a cell wall. The cytoplasm of the plant cell is partitioned from the inside out by the formation of a new cell membrane, called the cell plate, between the two daughter nuclei, rather than a contractile ring separating it from the outside in. Cell plate development includes the following phases: 1. Forming of the phragmoplast, a microtubule collection that directs and facilitates the formation of the cell plate. 2. Trafficking of vesicles to the division plane and a fusion to produce a tubular vesicular network. 3. Subsequent fusion of membrane tubules and a conversion into membrane sheets after callose deposition, accompanied by deposition of cellulose and other cell wall elements. That a higher plant cell has been dedicated to differentiating in a specific plane. A network of microtubules and actin filaments forms a ring around the whole cell just under the plasma membrane at this moment. The prophase band is named for the cytoskeletal sequence that occurs before prophase begins. As the cell moves to prophase, the band grows thinner and eventually disappears before metaphase. Nonetheless, the division plane has been established. As the new cell plate develops later during cytokinesis, it expands outward to unite with the parental wall precisely where the prophase band used to be, and acts as a track for vesicles to travel to the phragmoplast midzone. Lipids, proteins, and carbohydrates are found in these vesicles, which are needed for the creation of a new cell boundary. The Golgi apparatus has been established as the source of these vesicles by electron tomographic experiments. However, other research has indicated that they can also contain endocytosed materials. The meiotic spindle isn't enough to establish the precise location and direction of the cell plate on its own. In G2, the cortical collection of microtubules disappears in preparation for mitosis, which is the first visible indication. Occurrence of mitosis without cytokinesis. Fig 2.97 Mitosis without cytokinesis in the Drosophila embryo A to form a large syncytium. The first 13 nuclear divisions occur synchronously and without cytoplasmic separation. In a mechanism known as cellularization, most of the nuclei migrate to the cortex, where the plasma membrane expands inward and pinches off to envelop each nucleus, leading to formation of individual cells. B. Prior to cellularization, a fluorescence micrograph with several meiotic spindles at metaphase in a Drosophila embryo. The centrosomes are stained red and the microtubules are stained green. Undergoes remodeling by processes that are less well developed than those governing cytoskeletal and chromosomal dynamics. Function of lipids in cell division. Membranes are mostly made up of lipids, with an average of 50 to 100 lipid molecules per membrane protein. Different organelles have different lipid compositions, which may affect lipid-protein interactions and/or the physical properties of membranes, such as membrane fluidity or thickness, that sustain organelle work. Lipids may also be transferred between organelles through membrane contact sites, which are rapidly becoming essential functional centers in cells. Several lipids are found in cell division structures like the cleavage furrow in the midbody, and they may play a role in cell division. Membranes are associated with several cell division proteins. Some are integral membrane proteins that span the lipid bilayer, while others interact with membranes peripherally through lipid domains such as plextrin homology pH domains that attach to the sphenocytides. Finally, certain cell division proteins are lipidated, which means they have a lipid moiety attached to them after they have been translated. The lipid structure and localization of lipids within dividing cells are closely regulated. For example, disrupting single lipid biosynthesis causes cytokinesis failure. The sphenocytides' functions in cell division are becoming clear, including their control of actin dynamics and involvement in midbody maturation. Plasma membrane dynamics. In dividing cells, the plasma membrane is tightly bound to the cortex and rearranges to promote cell surface extension, follow various curvatures, and respond to mechanical stresses. Cells quickly change their volume and surface topology during mitosis. Since there isn't enough time for de novo membrane synthesis to handle these improvements, cells must rely on membrane shaping, distribution, and removal instead. Internal stocks may be replenished by trafficking activities such as exocytosis or endocytic recycling, or by plasma membrane remodeling, such as membrane gloving. During anaphase, there are exceptions to the rule that nuclear division is preceded by cytoplasmic division. Many cells go through several rounds of nuclear division with no cytoplasmic division in between. The first 13 rounds of nuclear division in a Drosophila embryo, for example, occur without cytoplasmic division, resulting in the development of a single large cell containing 6,000 nuclei arranged in a monolayer near the surface. Since the cells do not have to go through any of the steps of cytokinesis at each division, this structure significantly speeds up early growth. Following these rapid nuclear divisions, cellularization occurs, in which cells are formed around each nucleus in a single round of orchestrated cytokinesis. The plasma membrane expands inward and pinches off to enclose each nucleus, forming contractile rings at the cell surface. In certain forms of mammalian cells, nuclear differentiation happens without cytokinesis. Multinucleation occurs in osteoclasts, trophoblasts, and certain hepatocytes and heart muscle cells, for example. Remodeling of cell organelles in cell division. The cell division mechanism must ensure proper partitioning of cellular components between daughter cells in addition to segregating duplicated genetic information. The meiotic spindle facilitates chromosome separation before condensing into a midbody that is detached during cytokinesis. This mechanism is known as cytokinetic abscission. To ensure the proper segregation of genetic material between daughter cells, these steps are both supervised and performed with high spatial and temporal control. To respond to internal and external signals, new cells must have functional signaling pathways, as well as perform a variety of other tasks necessary for survival and development. Many of these roles take place in membrane-bound organelles, necessitating pathways to ensure that they are passed down properly. In most cases, de novo organelle synthesis will be inefficient due to the time and resources required, along with the potential risk of disproportionate organelle complements through daughter cells. During division, nearly every cellular organelle of the plasma membrane expands, allowing the spindle to be properly positioned for symmetric separation. The relationship between plasma membrane topology and spindle positioning tends to be reciprocal, with perturbation of spindle positioning leading to an attempt to recenter the apparatus through asymmetric cortical membrane expansion, possibly via membrane gloving. The plasma membrane is where the endocytic network begins. It's been questioned whether endocytosis occurs during cell division. Some researchers find that it doesn't, whereas others discover that endocytic recycling can be a subtly membrane for plasma membrane development. The partitioning of various endocytic cargoes that are essential to the cell, such as growth factor receptors, can be prioritized. Furthermore, various endocytic vesicles play essential roles in the later stages of cytokinesis. Figure 2.98 Overview of organelle dynamics during division. During anaphase, cytokinesis starts and concludes with abscission. During mitosis, the plasma membrane undergoes significant changes in structure. The nuclear pore complex disintegrates during prophase, and the nuclear envelope separates from the nucleus and merges with the endoplasmic reticulum. After that, the organelle is partitioned and passed on to daughter cells. During prometaphase, the Golgi is disassembled into vesicles, which are separated during metaphase and then reformed during telophase. Mitochondria fuse in interphase, fragment in prophase during meiotic entry, and fuse in anaphase. Prior to cytokinetic abscission, recycling endosomes aggregate at the spindle pole, and a subset of endosomes RAB1-1-FIP3 positive and RAB35 positive endosomes localized to the midbody. Protein LNP, which stabilizes tubular or network three-way junctions. As the nuclear envelope disengages from chromatin and nuclear envelope proteins are redistributed into the meiotic core, CDK1 dependent phosphorylation of INM chromatin and INM lamina tethers occurs. The link complex disengages from the lamina as a result of CDK1 dependent phosphorylation of the internuclear membrane protein SUN1. Inactivation of the phosphatidic acid phosphatase, lipin, inhibits de novo or membrane development and leads to nuclear envelope disassembly. Individual nuclear envelope and ER identities are destroyed after nuclear envelope breakup, and nuclear envelope proteins are partitioned into daughter cells alongside their elements. Golgi apparatus. As a single copy organelle, the Golgi must be disassembled and reassembled in daughter cells during separation to ensure equal inheritance. In the late G2 phase of the cell cycle, Golgi disassembly begins. The Golgi ribbon is converted into isolated mini-stacks where an ADP ribosylated substrate, CTBP1 bars, divides contacts between cisternae stacks. RAS can be phosphorylated by CDK1, PLK1, and NAP during meiotic entry to prevent transleadomerization and help Golgi ribbon unlinking. RAS65 also stabilizes microtubules, which aids Golgi clustering in interphase. Upon meiotic entry, it is phosphorylated and activated, which leads to ribbon unlinking by suppressing the cytoskeleton connection. At the G2M transition, Golgi disassembly happens in tandem with global cellular reorganization events like spindle assembly. To stimulate microtubule polymerization, spindle assembly factors such as TPX2 and activated Aurora A on microtubules. The connections between the plasma membranes, lipids, and proteins in the cortex are strongly ordered, coordinated, and limited. Physical restrictions are functionally important. For example, a diffusion barrier has been suggested in the cleavage furrow, which is based on filament-forming septin proteins that attach to both membranes and the cytoskeleton. Aniline and the esrin, radixin, moisin, and proteins are among the membrane-binding adapters that link the plasma membrane to the cell cortex in dividing cells. The small GTPase row and its effectors, as well as other key regulators of cytoskeletal dynamics, interact with the plasma membrane, and this association is needed for their work. Reversible interactions between the membrane and the cortex may help the dividing cell regulate and conduct membrane rearrangements with high temporal and spatial precision. ER and nuclear envelope. The endoplasmic reticulum is a network of membrane sheets and tubules that regulate secretory and transmembrane protein folding, sorting, and transport, as well as acting as a releasable store for intracellular calcium and a site for lipid biosynthesis. The ER and nuclear envelope have distinct morphologies throughout interphase, with membranous sheets surrounding the nuclear envelope and several morphometric proteins leading to the peripheral ER cisternae and tubular reticular layout. These proteins include reticulin, RTN1, RTN4, and receptor expression enhancing proteins, REAP1, REAP6, which stabilize strongly curved or tubules in the curved edges of sheets. Atlastins, a family of dynamic related GTPases that control homotypic fusion events, and Luna Park. Figure 2.99 Golgi rearrangement during cell division. In the transitional period, Golgi is made up of stacks cisterna connected by cisterna tethering proteins like Ras55 and Ras65, as well as Golgi proteins like GM130 and its adapter P115, which provides stability and aid in the capturing of incoming transport vesicles. The small GTPase R1 binds membranes in its active state and helps recruit the codomer CoB, not shown to aid in the formation of transport vesicles. Beat kinesis triggered in M phase phosphorylate Golgi localized targets such as GM130, Ras55, and Ras65 during division. This phosphorylation prevents Rafts from homodimerizing in separate spogons like GM130 from interactions with Rafts and tethering factors like P115. These changes result in the unlinking and disassembly of Golgi stacks, which helps to suppress incident vesicular flow. CTBP1 bars, a membrane fission protein, divides the Golgi into discrete mini-stacks, and GTPases like ARF1 inhibit Golgi structure, identity, and function. The development of small tubular vesicular clusters, known as Golgi haze, is caused by a block in vesicle fusion, allowing Golgi membranes to be acquired by stochastic processes or by interaction with the meiotic spindle. The meiotic Golgi protein GM130 also plays a role in steering spindle assembly by sequestering important and releasing its inhibition of the spindle assembly factor TPX2. Mitochondria. Mitochondria are double membrane organelles that generate ATP through oxidative phosphorylation and function as the primary energy producers in cells. Mitochondria undergo constant fusion and fission to conform to the cell's energy needs, and they can be transferred to areas of the cell with high energy demands through interactions with the actin and microtubule cytoskeleton. Mitochondrial fragmentation can assist stochastic mitochondrial inheritance during division. For example, mitochondria are moved to the poles of dividing cells and dispersed just before division in proportion to the divided cytoplasmic volume in Schizosaccharomyces samba. Nevertheless, stochastic distribution is unlikely to be the sole cause of mitochondrial segregation. Additionally, the centromere protein Fc and PF and the mitochondrial GTPases NEURO1 and NEURO2 bind mitochondria to microtubule tips during mitosis. Meiotic kinesin KIF5B member of the kinesin 1 family uses astral microtubules to transport mitochondria to the furrow during meiotic departure, similar to NEURO1, and meiotic phosphorylation induced decoupling of motor proteins from mitochondria helps symmetric inheritance of these organelles. NEURO1 and NEURO2 connect the mitochondria to microtubules in the actin cytoskeleton, according to studies in mouse embryos where the genes encoding NEURO1 and NEURO2 were knocked out. These connections will occur through the non-processive myosin motor unorthodox myosin 19, encoded by MYO19, which is located in the mitochondrial outer membrane. This protein is needed for symmetric mitochondria distribution between daughter cells, and its absence causes mitochondria to accumulate at spindle poles during meiotic departure, resulting in cytokinesis failure. Endosomes and other small organelles in addition to the main organelles mentioned above, cells use a variety of smaller membrane-bound structures, some of which have yet. At the G2M transition, Golgi disassembly happens in tandem with global cellular reorganization events like spindle assembly. To stimulate microtubule polymerization, spindle assembly factors such as TPX2 and activated Aurora A on microtubules. Assembly factors are hindered in interphase due to interactions with their nuclear localization sequences and importance. The phosphorylation of GM130 by CDK1 during meiotic entry disrupts its interaction with P115, exposing a nuclear localization signal within GM130. Importance is relocated to Golgi membranes by this nuclear localization signal, which relieves its suppression of TPX2-dependent spindle assembly. After being released from GM130, P115 will bind to spindle poles and assist in spindle assembly. As a result, Golgi disassembly is crucial for the creation and maintenance of the meiotic spindle. Between anaphase and cytokinesis, the Golgi regenerates quickly. To make the reformation of the Golgi cisterna, restacking of the cisterna, and the compression and fusion of stacks to form ribbons, regeneration necessitates the restoration of vesicular fusion. Dephosphorylation of GM130 by PP2A restores P115 dependent vesicular tethering and fusion, while dephosphorylation of BCI P135 restores its duty kinase function, restoring P97 mediated membrane fusion. In each daughter cell during telophase, twin Golgis form. A smaller twin develops near the cleavage furrow, while a larger twin forms near the centrosome. The smaller twin migrates across the nucleus during cytokinesis to coalesce with its sibling to regenerate a single Golgi. The explanation for this placement is unknown, although it's likely that the smaller twin supplies the midbody with Golgi-derived vesicles for abscission. The ARF1 guanine nucleotide exchange factor GBF1 was recently shown to be required for migration and coalescence of these Golgi twins, and inhibiting GBF1 degradation resulted in persistent Golgi membranes within the midbody, indicating that spatiotemporal regulation of Golgi reassembly is a key feature of meiotic escape. They are unusual to membrane-bound organelles in that they are surrounded by lipid monolayer rather than a bilayer. Lipid droplets can serve as reservoirs for membrane expansion and energy production, and their biogenesis and turnover are tightly controlled. Peroxisomes are oxidative organelles that participate in the synthesis of fatty acids and ether lipids as well as breaking down long-chain fatty acids by oxidation. They can be created from scratch and are replicated by division. During mitosis, peroxisomes congregate around spindle poles to promote symmetric inheritance. Loss of the peroxisomal protein PEX1-1B results in uneven peroxisome inheritance, misaligned meiotic spindles, and irregular differentiation of epidermal progenitor cells. Mitosis was also disrupted when PEX1-1B was forced to localize to indirect areas of the spindle using an optogenetic system, meaning that these small organelles play an important role in meiotic progression and that cells track and correct their segregation efficiency during cell division. Midbodies. Abscission, which takes place in the midbody and necessitates the collaboration of membrane and cytoskeletal remodeling operations, is when cell division is accomplished. Cortical ingression, the transition from a contractile ring to a midbody ring, and the preparation of the abscission site all require actin remodeling. Phosphatidylinositol 4, 5-bisphosphate PTDINS4, 5P2, a lipid that is found in the cleavage furrow, is a key regulator of actin dynamics. However, as cytokinesis progresses, PTDINS4, 5P2 is consumed, which is needed for furrow stability. RAP35 and 27-kDa inositol polyphosphate phosphate interacting protein organize the PTDINS4, 5P2 phosphatase OCRL of the midbody IPIP27. OCRL requires midbody actin to be cleared for effective abscission by hydrolyzing PTDINS4, 5P2. RAP35 also associates with, recruits, and stimulates the oxidative reductase nickel-1, which leads to actin clearance by depolymerizing actin filaments. Nickel-L1 transports RAB11 positive endosomes, and nickel-3 tethers. To be discovered biological functions. Since these small organelles are always multiple and complex, their inheritance can be less closely regulated than that of large single copy organelles. Endolysosomal network sorting and transporting intracellular cargoes to various subcellular destinations is the job of the endolysosomal network. While recycling, endosomes cluster around spindle poles and help coordinate spindle structure, which may assist their inheritance. The small size of these organelles suggests a stochastic model for their inheritance. Clathrin, which is best known for its interphase membrane trafficking functions, also aids meiotic spindle assembly. Meiotic exit necessitates the presence of endosomes. The small molecule prozosin inhibits endosomal sorting, resulting in cytokinesis failure, and the midbody contains a number of endosome-like membranous tubular vesicular structures. Exocyst-dependent vesicular fusion was once thought to be a mechanism for completing abscission, but more recent data indicate that endosomes that are functionally positive for RAB11 family interacting protein 3, RAB11 FIT3, or RAB35 supply proteins are needed for cytokinesis completion. When midbodies age, the frequency of intramidbody vesicles declines, implying that the need for these fusion events decreases as cells reach abscission. RAB11 FIP3 positive and RAB35 positive endosomes, on the other hand, accumulate near the potential abscission site and are used to produce a secondary ingression, an intramidbody structure that determines the final cut site. Autophagosomes the cell must eliminate and/or recycle post-abscission midbody residues after it has undergone division. Autophagy has been suggested as a mechanism, although some evidence suggests that the process is accomplished by phagocytosis. More study is needed to determine the specific functions of autophagy and autophagosome partitioning during separation. Lipid droplets are membrane-bound organelles that serve as storage organelles for neutral lipids and cholesterol. Intercellular nucleus displacement, especially cytomixis, is a special case of nuclear migration seen in generative plant and animal tissues, epidermis, and cell culture in vitro. Under some circumstances, it's unclear how and why the largest cellular organelle, which is stabilized by a constitutive network of structural proteins, will change shape and escape into small intercellular channels. Nucleus migration is commonly associated with a single occurrence in the organism's cellular life cycle, such as mitosis, meiosis, or fertilization, as well as cell division and polarization. The discovery of the linker of nucleoskeleton and cytoskeleton linked complex in the nuclear envelope, which mediates links to both MTs and actin filaments, has been an exciting development in recent years. Cash-Clarsic, ANC1, enzyme homology proteins in the outer nuclear membrane and SUN, SAD1 and UNC84 proteins in the inner nuclear membrane make up link complexes. All are type 2 membrane proteins with a single transmembrane fragment. At their C-terminus, cash proteins have a 60 residue cash domain that contains the transmembrane portion and up to 30 residues that project into the paranuclear region between the inner and outer nuclear membranes. Fungi and plants have less conserved cash domains than metazoans. The paranuclear space of SUN proteins has a conserved SUN domain. In humans, 5 genes code for SUN proteins, but only 2, SUN1 and SUN2, are commonly expressed. Lower eukaryotes have 1 or 2 SUN proteins. The diffusional abilities of the Cash and Sun proteins are close to those of other nuclear membrane proteins, meaning that they are possibly in dynamic complexes. Torsion is a possible link complex regulator since it is used in near lumen and paranuclear space and has an affinity for Cash domains. Torsion similarity to AAA acids means that it can help link complexes assemble or disassemble. The N-termini of Cash proteins, which vary in size and ability to bind cytoskeletal components, decide the specificity of link complexes. Sun proteins bind to nuclear lamins on the link complex's internuclear side. The nuclear lamina, a mesh under the internuclear vesicles within the midbody. Other nickel family members help coordinate vesicles within the midbody. 4.14. Nuclear migration. The nucleus is the largest organelle and the cell and is usually represented in the center. However, it shifts to an asymmetric location associated with cell function during cell division, migration, and differentiation. Depending on the cell form, cell cycle level, migratory state, and differentiation status, nuclei are often placed asymmetrically. Nuclei are transferred into the bud neck during cell division in budding yeast, for example, so that each daughter cell receives one. The nuclei of the fission yeast S palm are actively located in the center, meaning that the division plane produces two identical daughter cells. Male and female pronucleic acids into each other and merge in the center of the zygote in fertilized mammalian and invertebrate cells, meaning that the resulting cell division produces two identical daughter blastomeres. Asymmetric divisions, which are common in early embryos and stem cells, often indicate nucleus prepositioning. The molecular toolbox for nuclear placement includes one cytoskeleton components and two nuclear envelope protein complexes, Fig 2.100. The nucleus is moved by forces produced by the cytoskeletal components. Attachment of cytoskeletal components to the nucleoskeleton is mediated by protein complexes that span nuclear membranes. However, in most cases, particularly in plants, the molecular mechanisms and functional significance of nuclear positioning and migration are unknown. Actomyosin contraction and activation of the microtubule MT motor associated with the nuclear envelope are thought to be the two major mechanisms that act on nucleus positioning and displacement in animal cells. Because of its poor content in the cell cytoplasm and high degree of proteolytic degradation, studying the roles of actin, one of the major cytoskeletal proteins involved in nuclear migration in plants, presents a variety of challenges. For this and other factors, knowledge of the cytoskeleton's function in nucleus displacement processes in plant cells is limited. Lamins. While some proteins and their Nestrin partners have increased membrane diffusional mobility in mammalian cells lacking A-type lamins, they still localize to the nucleus. This means that link complex anchoring is influenced by other influences. While yeast lacks lamins, the nucleus is still attached to the cytoskeleton by Cash and Sun proteins. The heterochromatin binding protein E1 anchors the Sun protein SAD1, which is part of the spindle pole body in S comba. SAMP1, the mammalian equivalent of LNA1, is found in membrane is made up of intermediate filament proteins that polymerize to form the nuclear lamina. The primary lamins found in differentiated mammalian somatic cells are lamins A and C A-type lamins, which are alternate splice isoforms of the same gene, and lamins B1 and B2. SUN1 and SUN2's N-termini bind to lamina and mediate their contact with the lamina. As a result, the link complex binds the nucleoskeleton to the cytoskeleton through Cash protein interactions with cytoskeletal proteins and Sun protein interactions with Fig 2.100 molecular toolbox for nuclear movement positioning. An idealized link complex in the nuclear envelope is depicted in this diagram. Suns from the inner nuclear membrane INM bind to Cash proteins from the outer nuclear membrane ONM in the paranuclear region. Cash proteins bind to cytoskeletal filaments such as MTs, actin microfilaments, and cytoplasmic intermediate filaments directly or indirectly. Suns bind to the nuclear lamina and metazoans. Other internuclear proteins bind to suns in yeast and plants. For comparison, a nuclear pore complex NPC is seen. B. Different shades of blue reflect trimeric Sun2 domains, while orange represents the Cash peptide. The orientation of the Cash peptide between neighboring Sun domains is seen in the structure. Both unicellular and multicellular go through mitosis. The bacterial version of mitosis is known as binary fission. This differs from meiosis in that bacteria have only one circular chromosome, unlike eukaryotic chromosomes, and is not found within a nucleus. Two diploid cells of the same genetic makeup are formed. Only sexually reproducing species are enabled to perform meiosis. Since archaea and bacteria don't do this, it's easy to believe that unicellular species don't reproduce sexually. However, there are exceptions. Under nutritional stress, budding yeast will produce haploid spores. 4 haploid cells are created, each with a different genetic material. Mitosis lasts only half as long as meiosis. Mammalian cells in culture can take up to 10 hours to complete a cell cycle, while budding yeast can take up to 80 minutes to complete a cell cycle, and bacteria can differentiate every 20 minutes. In different species, meiosis occurs on different timescales, which are influenced by a variety of factors such as the organism's temperature and climate, as well as the amount of nuclear DNA present. Due to a developmental lock at prophase I, the process in yeast can last 6 hours, but in human females, it can last more than 40 years before ovulation. Other cases are 1 to 2 days in male fruit flies and 24 days in human males. In cancer, either tumor suppressors are turned off or oncogenes promoters of cell division are overactive, resulting in uncontrolled mitosis. Aneuploidy occurs when the incorrect number of chromosomes shows up in germ cells as a result of errors in meiosis. This can lead to miscarriage, but can also be inhibited or retained. Down syndrome, which is caused by trisomy of chromosome 21, is one example. Klinefelter syndrome, in which XY males have an extra X chromosome, is another example. Link complex assemblies that link actin to the nucleus. Amarin is an essential protein that binds to lamins and nesprins and is primarily found in the internuclear membrane. Sun1 also forms complexes with nuclear pore complexes. While the components of the link complex are the most important instruments for linking the nucleus to the cytoskeleton, they may not be the only ones. At the cytoplasmic phase of the nuclear pore complex, dynein interacts with the caudal 2, which then binds to RA-NBP2. During G2, this interaction directs dynein to the nucleus, perhaps contributing to nuclear envelope breakup. It could, however, be a different way to target dynein for nuclear movement. Certain nuclear membrane proteins found in muscles aggregate along MTs, implying that the nuclear positioning toolbox can also contain tissue-specific tools. 4.15. Comparison of meiosis and mitosis. Mitosis is derived from the Greek term mitos, which refers to the thread-like chromosomes observed under a microscope in dye-stained cells during cell division. Walter Fleming first discovered it in his 1882 work, Cell Substance, Nucleus, and Cell Division. In Greek, meiosis means lessening. As a result of meiosis, each new cell's genetic material becomes half of what it generally was to begin with. In 1876, Oscar Hertwig was the first to characterize the fusion of egg and sperm in a translucent sea urchin egg. Mitosis is a process that allows a unicellular organism to reproduce as a species. The aim of growth in a multicellular organism may be to expand during development or to restore ill damaged tissue. In preparation for sexual reproduction, meiosis creates gametes with just one copy of the organism's genetic information. Different stages of meiosis allow for genetic variation in the daughter cells. This is the starting point for evolution. Eukaryotes, phases meiosis, mitosis. Prophase, metaphase, anaphase, and telophase occur twice in meiosis. The first round of division is special, but the second round resembles mitosis more closely. Prophase, metaphase, anaphase, and telophase occur only once in mitosis. Prophase, the centrosomes tend to form an early spindle as the chromosomes condense. The first prophase of meiosis is much longer than the first prophase of mitosis. During prophase I, homologous chromosomes make chiasmata, or interactions with each other, and crossing over happens. This is where chromosomes trade DNA segments. This is important not only for producing genetic diversity but also for holding homologous chromosomes together mechanically. The centrosomes start to form an early spindle as the chromosomes condense. In comparison to meiotic prophase I, meiotic prophase is much shorter. In mitosis, there isn't any crossover. Metaphase, the action of microtubule spindle fibers originating from the centrosome situated at opposite cell poles causes chromosomes to line up around the metaphase plate during metaphase 2 of meiosis and metaphase of mitosis. Kinetic force of the centromeres of the chromosomes bind these fibers to the chromosomes. Pairs of homologous chromosomes line up around the metaphase plate in meiotic metaphase I. The law of individual assortment guarantees a spontaneous and independent distribution of chromosomes to the daughter cells of meiosis and eventually to the haploid gametes at the end of meiosis 2 by orienting homologous pairs randomly with respect to the cell poles. A single chromosome pair of chromatids lines up along the metaphase plate during meiotic metaphase. Since sister chromatids are similar, the chromosome's orientation has little impact. Anaphase, since the centromeres of the chromosomes are not cleaved during anaphase of meiosis, sister chromatids are kept together as homologous chromosomes are separated to opposite cell poles. The cohesin protein that holds the centromeres of the sister chromatids together is cleaved during anaphase of mitosis, and meiosis 2 causing the sister chromatids to segregate to opposite poles of the cell, where they are named chromosomes. Phases meiosis, mitosis. Telophase of freshly divided chromosomes continue to uncoil and become less condensed as the nuclear membrane reforms around them. The microtubules in the spindle dissociate. One centrosome will be passed on to each daughter cell. Cytokinesis of plasma membrane of the cell pinches, resulting in two daughter cells with distinct plasma membranes. Cytokinesis is needed twice during meiosis, once after telophase I and again after telophase 2. Cytokinesis does not necessarily occur in mitosis. Certain cells, such as muscle cells, differentiate and become multinucleate. Table 2.10 comparison between stages of meiosis and mitosis. Female and male meiosis. Meiosis is strongly sexually dimorphic, despite the fact that it is needed for the development of both eggs and sperm. Indeed, there is a case to be made that the discrepancies in male and female meiosis outnumber the parallels. Oocytes begin meiosis in the fetal gonad, enter an extended rest period (dicta) and do not restart meiosis until 10 to 50 years later, at the time of ovulation. Just a few hundred mature eggs are produced during a female's reproductive. Oocytes begin meiosis in the fetal gonad, enter an extended rest period (dicta) and do not restart meiosis until 10 to 50 years later, at the time of ovulation. Just a few hundred mature eggs are produced during a female's reproductive lifetime, and since sperm penetration stimulates the completion of the second meiotic division, only eggs that are successfully fertilized complete meiosis. In the male, on the other hand, meiosis begins with sexual maturation. Germline stem cells ensure that large amounts of sperm are generated continuously, and the meiotic period is relatively brief, about 3 to 4 weeks out of the 9 weeks needed to create a mature sperm cell. Males and females in mammals produce different numbers of viable gametes as a result of meiosis. Each spermatogonium produces 4 haploid spermatids of identical size in males. Females, on the other hand, have rather asymmetric cytoplasmic divisions during meiosis. Oocytes in the ovary are much larger than sperm when fully grown, and the potential egg holds the majority of this amount as it goes through meiosis. As a result, from each female meiosis, only one viable oocyte is obtained. The oocytes remaining 3 haploid cells are pinched off as polar bodies with very little cytoplasm. In addition to these apparent sex-specific temporal variations, there is a significant difference in error rate between the sexes, especially in humans. An approximate 5 to 10% of scientifically recognized human pregnancies are chromosomally defective, with the vast majority of these pregnancies ending in miscarriage or birth defects. Trisomy is the most common chromosome abnormality, which occurs when segregation errors occur during one of the 2 meiotic divisions. Non-disjunction can occur during spermatogenesis or oogenesis, but the vast majority, 75 to 90%, of trisomies are caused by errors during maternal meiosis, according to research. While the mechanisms underlying the high rate of errors in female meiosis are unknown, recent research suggests that several factors, including meiotic protein degradation and checkpoint stringency, can play a role. The only molecular mechanism that has been specifically related to human meiotic errors is meiotic recombination, which happens during the fetal stages of oogenesis. The failure to recombine genes across or initiate crossovers in genes in humans can lead to both paternally and maternally related VIGA. Chromosome misregulation is predicted to occur in just 1 out of every 10,000 meiotic cases. Since the essential meiotic pathway is heavily conserved across organisms, the underlying cause for the increased occurrence in humans is unknown. Meiosis is a mechanism that involves one round of DNA replication followed by 2 cellular divisions to produce haploid gametes. The first cell division, meiosis I divides homologous chromosomes, while meiosis II separates each homolog sister chromatids. Non-disjunction can occur at each of these meiotic stages of division. This is when polymorphic genetic markers are prominent at around the centromere of the non-disjoint chromosome. They can also differentiate the 2 options. As a result, a variety of aneuploid conditions have been investigated in order to ascertain the non-disjunction error's parental origin and meiotic stage. Monosomic disorders, which tend to cause early embryonic lethality, have a scarcity of information. Many trisomy complications, on the other hand, are compatible with at least some fetal growth, and outcomes with some trisomies are available. The first molecular correlate of human non-disjunction has recently been established as altered genetic recombination. Non-disjunction is linked to mutations that influence the amount and position of meiotic recombination in model organisms. In humans, it seems that changing the quantity and location of recombination raises the likelihood of non-disjunction. It is possible to recreate the recombination patterns that exist during meiotic events that result in non-disjunction. For all meiosis I trisomies studied to date, including maternally derived cases of trisomies 15, 16, 18, 21, and sex chromosome trisomies, studies of this kind have found substantial reductions in recombination. Trisomies with a correlation is especially strong in cases of maternal origin. At least 2 lines of research suggest that events arising during the fetal stages of oogenesis play a role in the development of human aneuploidy. First, research on ultrasonic pregnancies has linked defects in meiotic recombination, a mechanism that occurs in the fetal oocyte, to the root of maternally derived cases of trisomies 13, 15, 16, 18, 21, 22, and sex chromosome trisomies. Second, recent cytological analyses of human packaging oocytes show that a large proportion of cells have non-recombinant bivalence, implying that certain oocytes are predisposed to non-disjunction from the start of meiosis. 4.17. Chromosomal abnormalities. 1. Meiotic non-disjunction. The incorrect isolation of chromosomes during meiotic or meiotic cell division is known as non-disjunction. Non-disjunction causes dose mismatch, resulting in 1 extra or 1 missing chromosome in a proportion of the cells. Non-disjunction during mitosis is normal in cancer cells, while non-disjunction during meiosis occurs in chromosomally unbalanced gametes. If an unbalanced gamete fertilizes, the developing embryo may be aneuploid, with 1 chromosome too many trisomy or 1 chromosome too few dysplasia of monosomy. In most cases, those embryos are not viable, which has significant health implications for humans. Aneuploidy is the most prevalent chromosome abnormality, observed in at least 5% of all scientifically recognized. It is also the leading reported cause of birth, death, and fetal wastage. Aneuploidy is the leading hereditary cause of mental disability and development impairment in conceptions that live to term, primarily trisomy for chromosomes 13, 18, and 21, as well as different variations of the sex chromosomes. Non-disjunction is very rare in most species, unlike humans. For Saccharomyces sere. Figure 2.101 daughter cells without chromosome numbers result from non-disjunction. In meiosis I, first row, meiosis II, second row, and mitosis, non-disjunction occurs when chromosomes do not divide evenly, third row. These uneven separations may result in aneuploids, or daughter cells with unusual chromosome numbers. Non-disjunction prevents a haploid gamete from receiving a chromosome during meiosis, so it joins with another gamete to create a monosomic zygote. As a product of non-disjunction, a gamete receives a complete homologous chromosome pair and joins with another gamete to form a trisomic zygote. 2. Chromosomal translocation. The exchange of genetic materials between 2 non-homologous chromosomes is known as chromosomal translocation. Reciprocal non-Robertsonian and non-reciprocal Robertsonian translocations are the 2 types of chromosomal translocations. Reciprocal non-Robertsonian translocation involves exchange of genetic segments from 2 non-homologous chromosomes, Fig 2.102. This form of chromosomal translocation may be balanced. The shared genetic material is fully functional, or unbalanced. The exchanged genetic material is either extra or missing. Figure 2.102 consequence of chromosomal translocations. A. Reciprocal translocation occurs as fragments from 2 non-homologous chromosomes are exchanged. B. Recombination between the coding region of one gene and the regulatory elements of another gene causes the former gene to be upregulated. Chromosome translocations occur when 2 genes coding regions cross, resulting in the creation of a chimeric gene, which is then converted to chimeric protein. Reciprocal translocation carriers are normally healthy and show no signs, with an occurrence of around 1 in 500. Nevertheless, carriers gametes are more likely to have unbalanced chromosomal translocations and genetic defects. Where the short arms of 2 chromosomes merge in the centromeric region, non-reciprocal translocations Robertsonian occur. The long arms of both chromosomes are lost, and the number of chromosomes is reduced. On average, 1 Robertsonian translocation is seen in every 1,000 newborns. There are 2 forms of chromosomal translocations that result in the deregulation of specific genes or the development of novel fusion genes, depending on the place. When the coding area of one gene recombines with a transcriptionally active promoter or enhancer region of another gene, the former gene is upregulated. TMPRSS2ERG and TMPRSS2ETD1 translocations, immature or ETD1 near the target genes, are examples of such cases. Due to the repositioning after the TMPRSS2 transcription start site, which is actively transcribed in prostate cancer, ERG or ETD1 genes are significantly upregulated. Another example is NYC gene chromatin rearrangement of promoter elements of IGH switch regions in B-cell tumors, which is a hallmark of diffuse massive B-cell lymphoma. Chromosome translocations occur as 2 genes coding regions cross, resulting in the creation of a chimeric gene, which is corals, anemones, hydras. Meiosis may be stopped at any point between the replication of both chromosomes in meiosis II. Female meiosis in humans is stopped at the conclusion of meiosis I when fertilized. Human eggs have twice as many chromosomes as the sperm that fertilizes them. Female meiosis is asymmetrical in all animals and plants, whereas male meiosis is symmetrical. In eukaryotes, the symmetry of male meiosis appears to be uniform, with all 4 meiotic products having the ability to move on to the next generation. Just one of the products of female meiosis, on the other hand, develops into a viable egg cell, possibly after multiple intervening meiotic divisions in plants, and has the ability to pass on the DNA to the next generation. Animals discard the remaining components of female meiosis as polar bodies. In certain species, one of the meiosis I products is discarded as a diploid polar body that never goes through meiosis II, while the other meiosis I product goes through a second reduction to form a haploid egg and a haploid polar body. Thus, in mammals, female meiosis produces 3, not 4, things, all of which degenerate but 1. 3 of the 4 haploid results of female meiosis are discarded as polar bodies in many flowering plants. In other flowering plants, the haploid organism retains 2 to 4 of the haploid products of meiosis, resulting in a dysphoric or tetrasporic female gametophytes. Also in dysphoric or tetrasporic gametophytes, only one of the 4 products of flowering plant female meiosis contributes DNA to the egg cell in subsequent generations. Some female meiosis products include the core cell, which develops into endosperm, and presumably the antipodal cells. Individuals of full automixis, i.e., central fusion and terminal fusion, in which 2 of the haploid products of female meiosis simultaneously merge to restore diploidy, are the only plants or animals that contribute more than one of the haploid products of female meiosis to the next century. Also, with this, then converted into a chimeric protein. The fusion protein can improve constitutive kinase activity, allowing cancer to develop and advance. The chimeric protein BCR-ABL is a constitutively active tyrosine kinase encoded by an in-frame fusion between the ABL gene on chromosome 9 and the BCR breakpoint cluster region gene on chromosome 22. Inhibitors that target BCR-ABL, such as Gleevec, have been used in clinical trials and have been shown to improve the survival rate of CML patients. The initiation of chromosomal translocations was thought to be a spontaneous phenomenon that occurred under growth pressure selection. Latest research into a variety of factors that influence the spatial similarity of chromatins, DNA double-stranded breaks, DSBs, and translocation breakpoints suggests that chromosomal translocation cannot happen by chance. Physical interaction of translocation partners happens before DSB development in the contact first paradigm, which assumes that broken chromatin is confined to a regional field. Translocations arise among broken chromosome ends that undergo large-scale motion inside the nucleus to achieve proximity between translocation partners in a breakage first model. Latest analysis using the TMPRESS2-word chromosomal translocation model framework follows all of these models. AR can facilitate the spatial proximity of TMPRESS2 and the ERG or ETB gene loci. Androgen agonists make it easier for DSBs to form at translocation sites. Wide regions of movement in the broken chromatin ends mean that free DSBs can travel easily in the nucleus. As a result, both spatial proximity and site-specific DSBs can occur at the same time, are necessary for chromosomal translocation formation, and are affected by the same factors. 3. Meiosis sex differences. Female meiosis is stopped and resumed only after a sperm nucleus has reached the meiotic female cell in all species except sea urchins and cnidarians, e.g., jellyfish. The end of division until the beginning of the isotope uptake into new DNA. The isotope used labeled the DNA only during interphase. Taking the different observations into account, Howard and Pelk concluded that the time taken for DNA synthesis was about 6 hours and the cells underwent the next division only after 8 hours of DNA synthesis. They anticipated that there are 4 phases in the cell cycle: a period of cell division or mitosis, G1 or the gap 1 phase, the S phase DNA synthesis, and G2 or gap 2 phase, Fig 2.103. Figure 2.10.3 diagrammatic representation of cell cycle by Howard and Pelk. The chronology follows G1, S, G2, M phase. Phases of the cell cycle. The cell cycle results in the generation of somatic cells and sex cells in eukaryotes. The cell cycle of eukaryotic cells, like the Saccharomyces cerevisiae, budding yeast, and Schizosaccharomyces pombe, fission yeast, has been researched to find that the time taken by their cell cycle is about 2 to 3 hours. The time duration of cell cycle varies from organism to organism. Example, for humans, the cell cycle is 24 hours. Intense method of self-fertilization. Female meiosis is asymmetrical in that only 2, not both, of the meiotic components can be passed on to the next generation. The asymmetry of female meiosis seems to be universal, with 1 but rarely 2 meiotic product having the ability to be passed on to the next generation. The eukaryotic cell cycle. Growth and reproduction are the main characteristic properties exhibited by all living things. As stated by Rudolf Virchow, omnis cellula e-cellula means all cells arise from pre-existing cells. All cells, either prokaryotic or eukaryotic, eventually reproduce and ultimately die. The reproduction process in prokaryotes is simple and it is more complex in eukaryotes. Initiated from a unicellular organism which adopts a synchronized method of cell division and cell proliferation, an abundant number of cells are formed with differentiation to form a complex organism. Thus, huge numbers of cells which are witnessed begin their life from a single cell which undergoes cell division to produce 2 daughter cells which in turn continuously divide producing a plethora of cells. The period that initiates with the process of mitosis and the production of 2 daughter cells is defined as cell cycle. This is a continuous and tightly coordinated process. Unlike prokaryotes, the eukaryotic cell cycle is a complex one, which consists of 4 different phases: increase in the size of the cell, DNA replication, distribution of the double chromosomes to daughter cells, and cell division. Discovery of cell cycle. Alma Howard and Stephen Pelk, in 1953, introduced the mechanism of cell cycle for the first time. They worked on the roots of bean Vicia fava L by growing them in 32p isotope media. It took about 12 hours from No. Cell type time taken by cell cycle. 1. Frog embryo cells 30 minutes. 2. Fission yeast 2 hours. 3. Budding yeast 1.5 hours. 4. Intestinal epithelial cells 12 hours. 5. Mammalian fibroblasts 20 hours. 6. Human liver cells 1 year, 300 to 500 days. 7. Skin cells 2 weeks. 8. RBC 120 days or 4 months. 9. HeLa cells 20 hours. 10. Drosophila embryo 8 minutes. Table 2.11 cell cycle time period for different cells. The cell cycle of eukaryotes takes place chiefly in 2 phases of mitosis or M phase and the interphase. Mitosis phase or M phase being the shortest phase of cell cycle is divided into 4 phases: prophase, metaphase, anaphase, and telophase. This nuclear division is subsequently followed by cytokinesis. Cytokinesis is the process of division of cytoplasm, which along with mitosis gives rise to 2 daughter cells. Interphase. The inactive phase or resting phase between 2 successive meiotic divisions is known as the interphase, which takes the longest time of the cell cycle, about 95%. Though considered an active phase, the interphase is the period that shows the highest molecular activity as both the events like cell growth as well as replication take place in this phase only. The interphase is further subdivided into 3 phases: G1 phase, S phase, and G2 phase. G1 phase. G1 phase or gap 1 is the period between mitosis and initiation of DNA replication, where the cell becomes metabolically active to prepare itself for the replication of DNA which occurs in the next phase. The different biomolecules like proteins, lipids, and carbohydrates in this phase lead to a growth in the size of the cell. Though replication does not take place in the G1 phase, the cell makes itself ready for the replication process by producing the different types of biomolecules which will be required during the replication process. The new cells formed during M phase have to grow, so most of the physiological activity of a cell occurs in G1 phase. Thus, it is typically the longest phase of the cell cycle. If we consider the cell cycle of humans, the G1 phase takes about 10 hours of the 24 total hours of the cell cycle. G0 phase or the quiescent phase commences after the G1 phase to ensure that S phase and M phase occur successively and only once per cell cycle. Attached at a point on the DNA in G1 and this marks the starting point of replication in S phase. In the S phase, they are removed once replication begins, so that DNA replication occurs only once. Cell growth, in addition to DNA replication, continues all through the S phase along with the proteins and enzymes necessary for DNA synthesis. At the end of S phase, the cell has double the DNA content as it had before the commencement of S phase. Now that the cell is done with the S phase, it proceeds towards G2 phase or gap 2 phase. G2 phase. The second gap phase or the G2 phase is the period of introspection where the cell again becomes metabolically active to produce the biomolecules or proteins required for the M phase or mitosis. At this point, the cell inspects if the growth of the cell is adequate and the DNA is flawless or needs DNA repair. If the cell on inspection finds that everything is okay, then it proceeds to the M phase, otherwise enters into a dormant stage, which is known as the quiescent stage G0 of the cell cycle and stays in that stage till induced to divide. M phase. The last phase of the cell cycle is the M phase or the meiotic phase. This phase is marked by the nuclear disintegration releasing the DNA, equal distribution of the DNA to the 2 poles of the cells followed by cytokinesis leading to the division of cells into 2 daughter cells. Early embryonic cells are exceptions and do not undergo G1 and G2 phases, rather go straight to the S phase. A number of experimental evidence of the cell cycle have been demonstrated using autoradiographic technique. Cell cycle distribution of DNA content flow cytometry is used to differentiate the cells which are in different phases of the cell cycle. For the analysis, the cells are usually absorbed and treated with a fluorescent dye, like propionate iodide pi or 4,6-diamino-2-phenylenediol DAPI, which are meant for staining the DNA. Flow cytometry detects the. So the G0 phase is considered a gap period where the cell checks if everything is okay and then decides if it should proceed to the next phase. If the cell finds that everything is satisfactory, then it proceeds to the next phase of the cell cycle, the S phase or the synthesis phase. The cell enters the S phase only if the DNA is intact and the cell growth is adequate in the G1 stage. The protein P16 is found to usually repress the initiation of the S phase. P16 is actually a tumor suppressor protein which is considered to be a cyclin-dependent kinase inhibitor and is instrumental in the regulation of the G1S transition of the cell cycle. The G1S transition is guarded by a checkpoint and a cell can overcome this hurdle and proceed to S phase only when it builds certain proteins which overcome the repression caused by the P16 protein. During this phase, synthesis of new DNA takes place. S phase. The total DNA content of a particular cell decides the length of the S phase of the cell cycle of that cell and the rate of synthesis of DNA is stable between species and cells. In a human cell cycle, cells generally take 5 to 6 hours to complete S phase. The S phase of the cell cycle takes place throughout the interphase just before the initiation of the M phase of the cell cycle. This phase is known to be responsible for the DNA synthesis. So the genetic content of the cell is doubled prior to the M phase so that there is abundant DNA to be distributed between the 2 daughter cells. The S phase starts only after the cell has passed the G1 checkpoint and is ready to hold double the DNA. The protein P16 stops the S phase until the G1 phase has been passed through. The P16 protein is a tumor suppressing protein that prevents the occurrence of cancers. It cancels the activities of cyclin-dependent kinases so that the cell halts in the S phase and does not proceed to the next phase. S phase marks the replication of DNA so that double the amount of DNA is produced before the cell is ready to distribute equal amounts of DNA to the daughter cells. The regulatory pre-replication complexes are phases of the cell cycle. If the cell is supposed to have undergone any kind of DNA damage, the cell cycle abnormalities can be shown on the DNA content frequency histogram, e.g., DNA damage which suspends the progression of cell cycle at specific checkpoints. The cell cycle arrest caused by the DNA abnormality may lead to either DNA repair or cell death by the process of apoptosis. In 1969, a research group from Los Alamos Scientific Laboratory from the University of California was the first to analyze the cell cycle by fusion staining technique. Ottar Christian from Harvard Medical School, in 1975, provided the first protocol for analysis of the cell cycle by using propionate iodide staining. Intensity of fluorescence of the stained cells in the DNA content of the cell is correlated with the phase of the cell cycle. As we know that DNA synthesis takes place in the S phase and the DNA content of the cell doubles during this phase, the intensity of the fluorescence will also double. The fluorescence intensity is lower in the G0 phase and G1 phase, but is more in the G2 phase as this phase comes after the synthesis phase. The DNA content identifies the cell cycle phase position in the G0, G1 phase versus S phase versus G2, M phase of the cell cycle. The cellular DNA content of individual cells is often plotted as a frequency histogram to provide information about the relative frequency percentage of cells in the major. Figure 2.1048 typical cell cycle distribution of DNA content analysis by flow cytometry. Based on DNA content in individual cells, a cell population and exponential growth status can be divided into three subpopulations: G1, G0, S, and G2, M. Experimental evidence 8. Different phases of the cell cycle. The first evidence was provided by Howard and Pelk in 1951-1953 who labeled the roots of Visiophaga with 32P and detected radioactivity in the hydrolyzed preparations of meiotic cells. They observed that the radioactivity labeling was very low initially, but after the passage of about 10 hours, a number of dividing cells were labeled. This led to the conclusion that after the meiotic phase, a period of about 10 hours was needed for the DNA synthesis to occur. Investigated the timing of DNA synthesis in conjunction with mitosis and concluded that the DNA synthesis takes place in the S phase. Furkin and Burley in 1958 also used thymidine 3H labeled autoradiography to confirm that the duration of the G2 phase was the same as analyzed previously by Taylor et al. Using different cells, scientists have established the same facts about the cell cycle. For E.G., Painter and Drew, in 1959, used autoradiography in HeLa cell cultures in Quassler and Sherman in the intestinal epithelium of the mouse. Finally, they proposed that the cell cycle could be split into four phases: M, G1, S, and G2, with different time durations in the cells of different species. The next evidence was provided by Taylor et al. in 1957, where they introduced tritiated 3H labeled thymidine in the culture medium of Visiophaga. The radiolabeled thymidine is taken up by the cells in the S phase during the replication of their DNA. They observed that the DNA content doubles during the S phase and 2N becomes 4N. Here again, they. Figure 2.105 diagrammatic representation of Howard and Pelk experiment on Visiophaga to analyze the cell cycle. One of the first genes identified as an essential regulator of the cell cycle in yeast is CDC2, CDC28. Experimental evidence X. Cell fusion studies for progression of the cell cycle. Cell fusion experiments with cultured mammalian cells evidence that the cell cycle is regulated by some factors. When interphase cells in the G1, S, or G2 phase of the cell cycle were fused to cells in mitosis, their chromosomes condensed and nuclear envelopes retracted. This finding reveals that some diffusible components in the cytoplasm are associated with early mitosis. Later, these factors were named as cyclin CDK complexes. When cells in G1 were fused to cells in S phase and the fused cells were exposed to radiolabeled thymidine, the label was integrated into the DNA of the G1 nucleus as well as the S phase nucleus, proving that DNA synthesis began in the G1 nucleus immediately after fusion. However, when cells in G2 were fused to S phase cells, no integration of labeled thymidine was found in the G2 nuclei. Thus, diffusible factors in an S phase cell can penetrate the nucleus of a G1 cell and stimulate DNA synthesis, but these factors cannot induce DNA synthesis in a G2 nucleus. Regulation of the cell cycle. It is extremely crucial to regulate the cell cycle wherein each event has to be monitored and timed according to the right instance of requirement. Each and every step is tightly coordinated and plays an influential role in the integrity and functionality of the cell. Thus, accurate control of the cell cycle is critical for deciding the size and shape of each cell. Each step of the cell cycle is controlled by a multifaceted network of signaling pathways. Thus, we need to control each step to ensure that no error in the genetic material is conveyed to the next generation. The cell cycle must proceed with a lot of vigilance so that the division of the cell is proper and follows a regulated cascade of events. If not, it will undergo continuous division resulting in a tumorous or cancerous growth. Thus, experimental evidence 6. Chronology of the cell cycle through G1, S, G2, and M phase cells are visualized under the microscope upon staining with DNA binding fluorescent dyes or with antibodies against microtubules. Thymidine analog, bromodeoxyuridine Purdue, is supplied to be incorporated into the newly synthesized DNA. Propynium iodide pi or 4 feet, 6 feet TIAMIDINO2 phenylenediol DAPI can also be introduced to quantify cellular DNA content in cells. Cell staining with anti-Purdue antibodies and PI reveals the G1, S and G2 plus M phases of the cell cycle. Differences in light scattering allow separation of G2 phase cells from M phase cells. Another method could be adopted to analyze G1, S, G2, and M phases using preparation of isolated nuclei. Flow cytometric DNA content, in combination with side scatter measurements, are performed to discriminate interphase cells G1, S, and G2 phases from cells in mitosis due to differences in the chromatin structure of both types of cells. The fourth approach is based on the bivariate analysis of DNA content and proliferation associated proteins. The expression of cyclin D, cyclin E, cyclin A, or cyclin B1 with respect to DNA content is analyzed. This technique allows differentiating. For example, G0 from G1 cells, identifying meiotic cells or the expression of other intracellular proteins relative to the positioning of the cell cycle. Regulation of cell cycle has also been studied in some mutations of yeast that inactivate genes encoding important elements of cell cycle control system. These genes are known as cell division cycle genes, CDC genes. Most provisional cell cycle mutations are temperature-sensitive mutations, in which mutant proteins are unable to function at high temperatures but could progress at low temperatures. A temperature-sensitive CDC mutant can be propagated at a low temperature and then raised to a higher temperature to switch off the function of the mutant gene. CDC mutants will become abnormally large as they continue to grow. However, non-CDC mutants, deficient in a step that is essential throughout the cycle for biosynthesis and growth, halt abruptly at any stage of the cycle. Serodicia. It is present in the late G1 phase, which commits the cell to proceed from G1 to S phase. It was observed that once the cells have passed the start point, they were committed to proceed to the S phase and were liable to undergo the next cell division cycle. As this regulatory point is very delicate, it needs to be regulated tightly and restricted by external signals, such as nutrient availability and cell size. It was observed that when there was a shortage of nutrients, the yeast stopped the cell cycle at the start point and went through a resting state or G0 phase rather than proceeding to the meiotic phase, Fig 2.106. Thus, we can conclude that the start point in the yeast cell cycle exemplifies the juncture where the cell decides if the nutrients available are enough for the cell to progress through the remaining of the cell cycle. During the mating of the yeast, the arrest of the cell cycle at start is caused by polypeptide factors that permit the haploid yeast cells to fuse with one another rather than proceeding to the S phase. Number of checkpoints and regulators that are to coordinate the activities of the cell during cell cycle. We can say that the cell cycle checkpoints are tools for inspection which supervise the arrangement, reliability, and commitment of the major events of the cell cycle. Cell growth and extracellular signals. The signals from the intracellular and extracellular environment are extremely instrumental in coordinating and regulating the different processes involved in the eukaryotic cell cycle. The intracellular signals include the instigations produced by cellular processes such as DNA replication, mitosis, and growth of cells, while the extracellular signals are provided by growth factors that directly influence the proliferation of animal cells. A number of successive points of control are witnessed that regulate the progression of cell cycle. The regulatory point, known as the start in the cell cycle, has been very much investigated in the budding yeast Saccharomyces. Figure 2.106 Regulation of the cell cycle of budding yeast. It was observed that when there was a shortage of nutrients, the yeast stopped the cell cycle at the start point and went through a resting state or G0 phase rather than proceeding to the meiotic phase. Skip the restriction point and get committed to the S phase and rest of the cell cycle even if the further induction by the growth is stopped. Now, if the conditions are not favorable, e.g., if adequate growth factors are not available in G1, then the cell cycle halts at the restriction point. Thus, the cell ceases its progression and enters a quiescent stage called G0. The cells in the G0 are designated as arrested cells which remain in that stage for long periods without perpetuating or proliferating. Though they do not undergo division or growth, they are still metabolically active. On instigation by external factors again, they regain the proliferating property and divide luxuriantly. The most fantastic example of this type of stimulation is observed in the case of skin fibroblasts of animal cells. When there is a wound in the body of any animal or human being, platelet-derived growth factors are released from the blood platelets during the process of blood clotting which signal the skin fibroblasts to proliferate and rush for the healing of the wound. Start point in the budding yeast is not only involved in the decision of progression from G1 to S phase but also regulates the DNA replication and cell division. The cell size of the yeast cells has to be tracked to regulate the cell cycle events as the daughter cells attain a size which is more than the mother cell and are then committed to divide. Therefore, it necessitates the need to monitor the minimum size of the cell before it can progress forward so as to pass the start point. The G1 phase of the cell cycle is a critical phase in which the proliferation of the cells is regulated. A crucial point of decision called the restriction point is present in the G1 phase of animal cells, just like the start point in the yeast. But its regulation is different compared to that in the yeast cell cycle. We know that the passage of the yeast cell through the cell cycle is controlled by the availability of nutrients, but in animal cells, it is regulated by the extracellular growth factors which direct the proliferation of the cell. So the animal cells, in presence of growth factors. Figure 2.107 Regulation of animal cell cycles by growth factors. The proliferation of most of the cells are controlled in the G1 phase, but there are exceptions in which the regulation is at the G2 level. E.g., in the fish and yeast, Schizosaccharomyces pombo or oocytes on the animal cells. Here, the control is exhibited in the process of transition from G2 to M depending upon the cell size and the availability of nutrients in case of yeast in contrast to animal cells where the stimulation is hormonal. Figure 2.108a Cell cycle of fish and yeast. Beat division of the yeast cells into the daughter cells. Cell cycle checkpoints. Imagine if a cell progresses to the next phase with certain blunders created during the previous phase. What will be the result? This is a very important fact to ponder over because it involves the genetic material. A huge disastrous injury will take place at a genetic level which will be transmitted to the next generation and that will be very unfortunate for the next generation cells. E.g., the chromosomes of the cell in the S phase condense due to the MPF activity when S phase cells are forced to enter meiotic stage by fusing it with another cell in mitosis. Sometimes there may be fragmentation of the replicating chromosomes or occurrence of anaphase prior to the attachment of kinetochores to microtubules of the meiotic spindle during metaphase resulting in developmental abnormalities and mental retardation due to non-disjunction. The daughter cells must possess the duplicate chromosomes of the parent cell. If the flaws in the duplication or distribution of the chromosomes are transferred to the fresh cell produced, it may have to bear the burden of mutations causing various genetic diseases. Thus, to avoid these kinds of mistakes, we need a strict visual checkpoint to check every event occurring in the cell cycle. A checkpoint is a juncture in the eukaryotic cell cycle at which the cell slows down and scrutinizes the products of a particular phase of the cell cycle and concludes if the cell should proceed to the next phase or not. In order to reduce such mistakes, the cell cycle is monitored at 4 key checkpoints. These checkpoints make sure that chromosomal aberrations are prevented and that each stage of the cell cycle is completed prior to the commencement of the next stage. These checkpoints occur near the end of G1, at the G2M transition, the SG2 transition, and during metaphase. Figure 2.109 the 4 checkpoints that control the passage through the cell cycle. The G1 checkpoint. The G1 checkpoint is there to monitor whether all the circumstances of the cell are positive for cell division to occur further. We designate the G1 checkpoint as G1S cell cycle checkpoint or restriction point, as in yeast cells. The entry of the cell from G1 phase to S phase of DNA synthesis is coordinated by this checkpoint and this is an irreversible event. The passage of the cell through this checkpoint is decided by the adequate size reached by the cell and the assets of energy that the cell possesses. The DNA damage of the cell is also investigated at this point and if all the factors are found favorable, then the cell progresses to S phase and if not, then the G1 checkpoint halts the cell in the G0 phase till all the discrepancies are rectified. Fig 2.110. There are various external factors like the growth factors which influence the event of the passage of cells from G1 to S phase. The cell signaling molecule cyclins and cyclin-dependent kinases CDKs signal the transition from G1 to S phase. Figure 2.110 the G1 checkpoint showing that DNA damage can lead to G1 arrest. The G2 checkpoint. The G2 checkpoint checks the entry of the cell from G2 phase into the meiotic phase if any faults are found in the replicated DNA. This checkpoint ensures that all DNA has been correctly replicated without any type of flaw. If the checkpoint detects any type of problem with the DNA, then it halts the cell cycle, providing a chance for the repair of the damaged DNA or complete the DNA replication. If the DNA is found to be correctly replicated and without any mistake, then the cyclin-dependent kinases CDKs prompt the cell to start the meiotic cell division. This checkpoint is crucial because it aids the cell to retain the stability of its genome and thus it can be used for understanding the molecular mechanism leading to cancer. Figure 2.111 the G2 checkpoint showing the different conditions for the cell arrest at the G2 phase. The M checkpoint. The M checkpoint, also known as spindle checkpoint, occurs in close proximity to the end of the metaphase stage of mitosis. This checkpoint ensures that all the sister chromatids are attached properly to the spindle fibers. During the metaphase, the microtubules forming the kinetochores pull the sister chromatids backward and forward so that they line along the equatorial plane. The checkpoint ascertains that kinetochores of each pair of sister chromatids are firmly anchored to at least 2 spindle fibers and then only allows the cell to proceed further. This step is also very crucial because the separation of the sister chromatids during anaphase is an irreversible process and so extra care must be taken so that the attachment of the kinetochore to the spindle fibers is firm and proper separation of the sister chromatids takes place. Figure 2.112 the M checkpoint or the meiotic checkpoint. The S arrest. When a cell is not able to duplicate its DNA, an event of arrest occurs in the S phase of the cell cycle. Cell fusion experiment by Rao and Johnson. It has been observed and established that the G2 checkpoint does not allow mitosis to begin until the S phase of the cell cycle is completed. This activity ensures that the incompletely replicated DNA is not transmitted to the daughter cells. Moreover, once the DNA has been replicated in the S phase, there must be some mechanism which prevents the initiation of another S phase prior to mitosis. Thus, the control mechanism stops the cells in G2 phase from again entering the S phase and blocks the initiation of another round of DNA replication until the mitosis phase has ended. The dependence of S phase on M phase was established by the cell fusion experiments performed by Kotu Rao and Robert Johnson in 1970, where they isolated and fused the cells in different phases of the cell cycle to form several hybrids. Fig 2.113. It was found that when cells in SG1, G2 are fused with a cell undergoing of the DNA of archaea as well as eukaryotic organisms, these proteins bind to the origins of replication with ORC, origin recognition complex. The ORC is a heteromeric, 6-membered protein moiety, which is required for eukaryotic DNA replication. The replication origin sites in the DNA are bound by the ORC proteins utilizing the ATP and this can be regarded as a scaffold for the assemblage of initiation factors. MCM proteins are considered as the positive regulatory factors required for the initiation of DNA replication. So we can say that the MCM proteins can only bind to DNA in G1, allowing DNA replication to initiate in S phase. Once the replication has been initiated, the MCM proteins are dislodged from the origin of replication site, so that the second round of replication does not take place again until mitosis is completed. Mitosis, the nucleus of the non-meiotic cells is motivated to perform mitosis. When G1 cells were fused with S phase cells, the G1 nucleus instantaneously started synthesizing DNA, which indicated that the cytoplasm of cells in S phase has certain factors which were able to coax the cells in G1 phase to synthesize DNA. This suggests that positive factors drive entry into S and M phases. Again, it was observed that when cells in G2 were fused with S phase cells, the G2 nucleus failed to instigate the synthesis of DNA. This exhibited that a mechanism existed which prevented the replication of the DNA until mitosis had completed perfectly. When explored further, it was found that a unique group of proteins, the MCM proteins, restricted the DNA replication to once per cell cycle. The MCM proteins categorize a group of about 10 conserved factors which aid in the replication. Figure 2.113 the cell fusion experiment by Rao and Johnson in 1970 showing the coupling of S phase to M phase. Thus, to conclude, we can state that regulation of the cell cycle involves processes which are very essential for the survival of the cell, which includes the detection of DNA damage and its repair and also the prevention of uninhibited cell propagation. With the help of the experiments performed with the cell fusion of oocytes at different cell cycle phases, the cell cycle regulatory mechanisms can be elucidated that control the order of molecular events in the cell cycle. Maturation promoting factor MPF, a dimer of CDC2 and cyclin. A well-known experiment by Rao and Johnson in 1970 mentioned above provided us with the example. All the events of M phase such as nuclear envelope breakdown and chromosome condensation are coordinated by M cyclins. G1 cyclins. Unlike the other cyclins, the concentration of G1 cyclins increases gradually all throughout the cell cycle depending on the growth of the cell and certain signals from the external environment. The growth of cells and entry into a new cell cycle is coordinated by G1 cyclins, also known as cyclin D. They do so by inducing G1S and S cyclins expression. The G1 cyclins complex with the CDKs and activate G1S cyclin CDK by increasing the transcription of G1S cyclin and removing an inhibitor of G1S cyclin CDK. So, G1 cyclins are treated as crucial for DNA replication and cell division. In embryonic stem cells and cancer cells, G1 cyclins are necessary for the activation of cyclin-dependent kinases in order to phosphorylate the core pluripotency factors and sustenance of the pluripotency of the cell. G1S cyclins instigate the early cell cycles proceedings such as doubling of the microtubule organizing center (MTOC) or centrosome spindle pole body or SPB in the eukaryotic cells like yeast cells. When there is an increase in concentrations of G1S cyclins, we witness the accumulation of S cyclins like CLB5 and CLB6 in budding yeast. These S cyclins form S cyclin CDK complexes that motivate the replication of DNA. The S cyclins. This is represented by cyclin A, which when binds to CDKs get activated and the complex straightaway induces the replication of DNA. The concentrations of S cyclins is high all through the S phase as well as G2 phase and also in early meiotic phase to help early events to occur in mitosis. Cyclins A1 and A2 are two types of S cyclins. The germ cell lineages witness the presence of cyclin A1 while cyclin A2 is articulated universally in all the cell types. The concentration of cyclin A2 is seen to be low at the G1S boundary and then it starts elevating with the commencement of DNA replication and then gets degraded in the late G2 phase. During the S phase, cyclin complexes with CDK2 thus. Concept of an unknown factor that could force frog egg cells to enter M phase. This unknown factor was discovered as maturation promoting factor MPF in 1980s, which contains CDKs bound to M cyclins. This complex of cyclins and CDKs or cyclin-dependent kinases work in coordination to regulate the cell cycle transformation. Similar to the characteristic of cyclin, the M cyclin are present at very low levels during most of the tenure of the cell cycle, but its concentration increases as the cell proceeds towards the transition of cell from G2 phase to M phase. Now, as the concentration of M cyclin increases and gets collected in the cell, it binds to cyclin-dependent kinases or CDKs which are previously present in the cell and assemble into complexes that trigger M phase. These complexes become active and promote the events of M phase when they receive the indication that the cell's DNA is perfect and intact. It was found that the MPF complexes phosphorylate the different proteins present in the nuclear membrane, consequently disintegrating and condensing the chromosomes. Families of cyclins and cyclin-dependent kinases. Cyclin is a family of proteins that regulate the succession of cells through the cell cycle as a result of the activation of cyclin-dependent kinases, CDK. In 1982, cyclins were discovered by R. Timothy Hunt during the study of cell cycle sea urchins. Cyclins were named so because their concentration during the cell cycle fluctuated cyclically. The cyclin complexes with CDKs to activate them and produce active sites which need further phosphorylation for working henceforth. No enzymatic activity is produced by the cyclins themselves, but they possess certain binding sites to which CDKs bind, thus endowing the enzymatic property to it. In eukaryotes, cyclins are basically of four types: G1 cyclins, G1S cyclins, S cyclins, and M cyclins. The names of the cyclins themselves reveal that each cyclin is associated with a specific phase of the cell cycle and coordinates the events of that particular period. For catalysts, the events like meiotic spindle assembly as well as alignment of sister chromatids along the spindles. When the assembly of spindle is adequate and checked by the checkpoint, then the degradation of M cyclins commences in metaphase and anaphase, causing the exit of the cell from mitosis and cytokinesis. Mammalian cyclins and CDKs are studied for understanding their functions, each defined by their association with the different stages of the cell cycle. Phosphorylating the substrates and initiating DNA replication. Cyclin E-CDK2 complex is also found to regulate the end of S phase by activating the meiotic cyclin CDKase complexes. M cyclins. M cyclins are also known as B-type cyclins. The concentration of the M cyclin starts increasing as the cells enter the meiotic phase and reaches the maximum concentration at the metaphase. These cyclins, when complexed with the CDKase. Figure 2.114 concentration of different cyclin proteins during the stages of the cell cycle. Cyclin-associated CDK functions of cyclin CDK complex. Cyclin E-CDK1, CDC2, CDK2S phase entry. Cyclin B1, cyclin B2. CDK1 G2 exit, mitosis M phase. Cyclin C-CDK G0 to S phase transition, RNA polymerase II transcriptional regulation. Cyclin D1, cyclin D2, cyclin D3. CDK4, CDK6 G0 to S phase transition, RBE2F transcriptional regulation. Cyclin E-CDK2 G1 to S phase transition, RBE2F transcriptional regulation. Cyclin G1, cyclin G2 CDK5 DNA damage response. Cyclin-associated CDK functions of cyclin CDK complex. Cyclin H-CDK7 DNA repair, CDK activation, RNA polymerase II. Transcriptional regulation, cyclin L-CDK11 RNA splicing, cyclin T1. Cyclin T2. CDK9 RNA polymerase II transcriptional regulation. Table 2.12 different types of mammalian cyclins and CDKs and their functions. Cyclin-dependent kinases. Cyclin-dependent kinases, CDKs, can be categorized as a family of protein kinases which have a very crucial role in the regulation of cell cycle. CDKs are comparatively small-sized proteins, having low molecular weights 34 to 40 kDa and a domain for kinase activity. Cellular processes like transcription, processing of mRNA, and the differentiation of specific cells are controlled by the activity of the CDKs. The kinase activity of the CDKs is aggregated when complexed with cyclins. About 9 CDKs are found in the animal cells numbering from CDK1 to CDK9. Out of the 9 CDKs, the first 4 CDKs namely CDK1, CDK2, CDK3, and CDK4 are directly associated with the cell cycle regulation. CDK1 of mammalian cells form complexes with cyclin A2 and B1 to regulate the passage of cell cycle while CDK7 operates as CDK activating kinase. The principal components of the cell cycle control system are cyclin-dependent kinases, CDKs. The cyclic changes in CDK activity are mainly governed by cyclins. CDKs are completely dependent on cyclin for their activity. They could exhibit function only if tightly bound to cyclin. Cyclins are periodically synthesized and degraded in each cycle. However, the levels of CDK proteins are almost constant in each cycle. Cell cycle regulatory proteins, CDK inhibitor proteins, CDKIs. Protein function. SYK1 is a stoichiometric inhibitor of CDK1-CLB B-type cyclins complexes in the budding yeast. It prevents premature S phase entry. P15P1 arrests the activation of the CDK kinases, CDK4, by cyclin D, thus the encoded protein functions as a cell growth regulator that inhibits cell cycle G1 progression. P16 slows down the cell cycle by inhibiting progression from G1 phase to S phase. P18 prohibits the activation of the CDK kinases, CDK4, CDK6, thus functioning as a cell growth regulator that controls cell cycle G1 progression. P19 restricts the activation of the CDK kinases, CDK4, CDK6, thus functioning as a cell growth regulator that controls cell cycle G1 progression. P21 to P21, CIP1, WAF1 protein binds to and inhibits the activity of cyclin CDK2, CDK1, and CDK4/6 complexes, and thus functions as a regulator of cell cycle progression at G1 and S phase. Cell cycle regulatory protein CDK inhibitor proteins, CDKIs. Protein function. P27 KIP1 inhibits the catalytic activity of CDK4 and subsequently prevents CDK4 from adding phosphate residues to its principal substrate, the retinoblastoma PRB protein. Increased levels of the P27 KIP1 protein typically cause cells to arrest in the G1 phase of the cell cycle. P57 encodes a cell cycle inhibitor that binds to G1 cyclin CDK complexes. Thus, P57 KIP2 arrests the cell cycle in G1 phase. Table 2.13 cell cycle regulatory protein CDK inhibitor proteins, CDKIs. Experimental evidence 11. Coordination between MPF activity and cyclin B level in cell cycle YY maturation promoting factors. MPF is a protein kinase that requires a meiotic cyclin for activity. The protein kinase activity of MPF triggers mitosis by phosphorylating multiple specific protein substrates. In an experiment, researchers tracked the MPF activity and concentration of cyclin B in the egg extracts of the species of Xenopus. MPF activity reached its highest levels in conjunction with high cyclin B concentration during the early events of mitosis that followed chromosome condensation and disassembly of the nuclear envelope. The introduction of cyclohexamide, an inhibitor of protein synthesis, prevented cyclin B synthesis and also disallowed the rise in MPF activity, chromosome condensation, and nuclear envelope disassembly. These studies revealed that MPF activity ripples in synchrony with the concentration of cyclin B. YY in another experiment, all mRNAs in the egg extract were degraded by treating with a lower concentration of RNAs followed by inactivation with a specific inhibitor that resulted in damaged mRNAs without affecting the turns in RNAs required for protein synthesis as their degradation required much higher concentrations of RNAs. Sperm nuclei were further added to the RNase treated extracts. The 1N nuclei replicated their DNA, but the increase in MPF activity and the early meiotic events, chromosome condensation and nuclear envelope disassembly, which were observed in the untreated extract, did not take place. YY the addition of cyclin B mRNA produced in vitro from clone cyclin B cDNA to the RNase treated egg extract and sperm nuclei causes an like momentum in the cyclin B concentration and MPF activity. The featured early and late meiotic events are noticed within the untreated egg extract. Synthesis of only cyclin B under these conditions proves that it is the crucial protein required to regulate the MPF activity. YY determined the correlation of degraded cyclin B with the exit from mitosis. A mutant mRNA encoding non-degradable cyclin B was added to a mixture of RNase treated Xenopus egg extract and sperm nuclei. It resulted in increased MPF activity along with the level of the mutant cyclin B, initiating early meiotic events, condensation of the sperm chromatin and nuclear envelope disassembly. However, the produced mutant cyclin B of this in vitro reaction was not degraded. Eventually, the MPF activity was enhanced, and the late meiotic events, chromosome decondensation and nuclear envelope formation were both halted. This study established that decreased MPF activity and exit from mitosis rely on the degradation of cyclin B. Figure 2.115 The synthesis and degradation of cyclin B influence the cyclic process of MPF activity and meiotic events in Xenopus egg extracts. In each case, the MPF activity and cyclin B concentration were estimated at different timescales following the addition of sperm nuclei to a Xenopus egg extract, treated as shown in each box. Microscopic findings prove the onset of early meiotic events, blue shading including chromosome condensation and nuclear envelope disassembly, and late events orange shading including chromosome decondensation and nuclear envelope reassembly. Studies with Xenopus egg extracts prove that cyclin B is modified by covalent attachment to multiple ubiquitin molecules that leads to their degradation. This process of polyubiquitination ensures proteasome-mediated rapid degradation of proteins in eukaryotic cells. The multisubiquin anaphase promoting complex (APC) is a ubiquitin ligase that identifies a conserved destruction box sequence in meiotic cyclins and allows their polyubiquitination, subjecting the proteins to rapid degradation by proteasomes. The decrease in MPF activity attributes to the completion of mitosis. The ubiquitin ligase activity of APC is well regulated so that polyubiquitination of meiotic cyclins occurs only during late anaphase. Inactivation of APC in G1 allows increased concentration of meiotic cyclins during the next cell cycle. This maintains the cyclic rise and fall in MPF activity that results into the entry and exit from mitosis. Operation of cyclin CDK complex is a complex of cyclin and CDKase act as regulators of the eukaryotic cell cycle and are found to be present in all known eukaryotes. Their chief role as regulators of cell cycle is witnessed to be preserved through evolution as they are found in many different types of species, from yeast to frogs to humans. These complexes phosphorylate the substrates in the cycle to activate them. Cyclin CDK complexes previously present in the cell cycle phase phosphorylate the cyclin CDK complexes which appear in the later phase and activate them. CDKs possess a very low kinase activity of their own but get activated when complexed with cyclins. CDKs are designated as serine and threonine kinases as they phosphorylate their substrates on specific amino acid residues, serines and threonines. The phosphorylation site in the amino acid sequence of a CDK, the button yeast Saccharomyces cerevisiae, discovered temperature-sensitive mutants (CDC) that were faulty in cell cycle progression and underwent growth arrest at particular points in the cell cycle. The CDC28 protein is needed to channel through this critical regulatory point in G1. Similarly, Palmers et al. Also worked on a collection of cell cycle mutants of fission yeast Schizosaccharomyces pomba. The mutants of fission yeast were identified as CDC2 mutants. These CDC2 mutants are capable of arresting the cell cycle both in G1 and at the G2 to M transition phases observed in organisms like S. pomba. The scientists revealed that S. cerevisiae CD28 and S. pomba CDC2 are homologous genes, which are essential for the passage through start as well as for entry into mitosis in yeasts. It was established that CD2 encodes a protein kinase that is involved in phosphorylation which helps to regulate the cell cycle. Substrate consists of ST-PXKR consensus sequence. The phosphorylated serine or threonine is represented as ST. P stands for proline, X is any amino acid, K is lysine, and R is arginine. Now the question is that how does this phosphorylation help? A very apt explanation to the question is that when the cyclin complexes to a CDK, two important consequences are witnessed. On one hand, it activates the CDK as a kinase, and on the other hand, it coordinates the CDK to a particular group of target proteins specific for a specific phase of the cell cycle. The cyclins of G1 S phase force the CDKs to S phase destinations promoting DNA replication, whereas the M cyclins trigger CDKs to M phase targets, breaking down the nuclear membrane. Regulation of cell cycle in yeast. In 1970s, Lee Hartwell et al. Studied cell cycle regulation in yeasts. They, while investigating. Figure 2.116, the temperature-sensitive CD28 mutant replicates normally at the permissive temperature. At the non-permissive temperature, however, progression through the cell cycle is blocked at start. As well as threonine 14, invertebrates. The tyrosine 15 phosphorylation, catalyzed by a protein kinase called B1, inhibits CDC2 activity and leads to the accumulation of inactive CDC2 cyclin B complexes throughout S and G2. The transition from G2 to M is then brought about by activation of the CDC2 cyclin B complex as a result of dephosphorylation of threonine 14 and tyrosine 15 by protein phosphatase called CDC25. Regulation of MPF in mammals. Synthesis of cyclin B in mammalian cells begins in the S phase. The accumulated cyclin B complexes with CDC2 in the S and G2 phase. With the formation of the complex, phosphorylation of CDC2 takes place at two critical regulatory positions. One of these phosphorylations occurs on threonine 161, which is needed for the activity of CDC2 kinase. The next phosphorylation occurs at tyrosine 15. Figure 2.117 MPF regulation. CDC2 forms complexes with cyclin B during S and G2. CDC2 is then phosphorylated on threonine 161, which is required for CDC2 activity, as well as on tyrosine 15 and threonine 14 in vertebrate cells, which inhibits CDC2 activity. Growth factors of the D-type cyclins among all the types of cyclins present in the cell. The D cyclins are one of the most investigated cyclins. D-type cyclins that include cyclins D1, D2, and D3 form the G1 cyclins. Although the genes of the D-type cyclins are present on different chromosomes, the proteins expressed by them are identical 57-62% and have 140 residues conserved in each protein. Only the presence of cyclin box domain is common in the D cyclins with other cyclins in the cell. The proliferation of animal cells is under the control of a number of external signals like growth factors that manage the succession of cells through the restriction point in the late G1 phase. When there is a deficiency of growth factors, the cells are unable to pass the restriction point and become quiescent, frequently entering the resting state known as G0. The growth factor signaling and cell cycle progression are closely interlinked, and this communication is provided by the D-type cyclin D complexes are commonly found in human cancer cells. Inhibitors of cell cycle progression. Several facts regarding the regulation of cell cycle were revealed on a tight control of the cell cycle. In the passage of investigating the uncontrolled cell cycle, some regulatory proteins were discovered whose functions were damaged due to some kind of catastrophe. So the non-functional proteins now could not control the events in the cell cycle, and thus the cells started replicating in an uncontrolled manner, leading to cancer. The regulation of cell cycle and the proliferation of cells not only involves the positive factors which induce the cell to undergo cell cycle progression, but some negative agents as well, which restrict the cell cycle at some particular stages. The external factors may include physical and chemical signals. The negative factors like the damage to DNA, cell contacts, and various other extracellular factors work together to hinder rather than motivate the proliferation of cells. The mammalian cells show the property of contact inhibition, i.e. they form a single layer in the substrate and stop dividing as soon as they touch other cells. Further proliferation of the cells is inhibited by the signals generated when the cells come in contact with each other. Such inhibitory signals are produced due to the induction of CDK inhibitors. DNA of a cell gets damaged if the cell is exposed to any carcinogen or UV rays from the sun. Role of p53 in cell cycle inhibition. Now let us understand the concept of how DNA damage is interlinked with the halting of cell cycle at G1 phase. As soon as the DNA damage occurs, a protein known as p53 starts to get expressed, which is instrumental in blocking the succession of cell cycle at G1 phase itself. This p53 is known as tumor suppressor protein, and the gene expressing it is called the tumor suppressor gene. It is also denoted as a guardian of the genome, as it keeps a strict vigil on the status of the genome and acts. Cyclins. When there is stimulation by a growth factor, the synthesis of cyclin D takes place by the RAS-RAF-ERK signaling pathway synthesis, and this induction continues till the growth factors are present there. As soon as the growth factors are removed, the D-type cyclins are also quickly degraded. So we can conclude that as long as growth factors are available in the surrounding, in the G1 phase, complexes of CDK4, 6 cyclin D force the cells through the restriction point in the cell cycle, and the cell proceeds to the S phase. Figure 2.118 effect of growth factors and D-type cyclins on cell cycle. Inversely, it was witnessed that if the growth factors are withdrawn from a key point of regulation in the cell cycle, the concentration of cyclin D decreases immediately, and so the cells are checked at G1 phase and are not able to progress through G1 to S. They become quiescent and enter the G0 phase. The D-type cyclins orchestrate the signaling by growth factors with the cell cycle machinery. This reveals that the availability of extracellular growth factors is effective in controlling the progression of cells through G1. Any shortcomings in the regulation by cyclin D would lead to the loss of growth regulation of the cells, resulting in cancer. For example, mutations resulting in continual and regulated expression of cyclin D1 lead to the development of diverse human cancers, including lymphomas and breast cancers. Similarly, mutations that inactivate the INK4 CDK inhibitors that bind to CDK4, 6 lower expression of the DNA damage to the next generation of cells. With the rise in the p53 concentration, the synthesis of p21 protein is stimulated. When the p53 imposes blockade in the cell cycle, p21 prosecutes the halt in the cycle dictated by p53 by binding to and inhibiting the activity of the CDK cyclin complexes. p21 binds to proliferating cell nuclear antigen pCNA, which is a subunit of DNA polymerase delta, thus blocking DNA replication. Fig 2.119. So, p21 is functional in arresting the cell cycle on the occasion of DNA damage, where it blocks the cell cycle progression by inhibiting the CDKs, and on the other hand, it can directly inhibit DNA replication in cells in the S phase. As the cell is exposed to more stress, higher levels of p53 and p21 accumulate, making it less likely that the cell will move into the S phase. Accordingly, the 53 mentioned refers to the functional molecular masses of the proteins P in kilodaltons. It ensures that no damaged DNA is approved for the next generation cell. The p53 protein has diverse functions. The first role it plays is in halting the cell cycle at G1 checkpoint via motivating the production of CDK inhibitor protein, which is abbreviated as CKI. These inhibitors bind to the CDK and cyclin complex and restrict the passage of the cell cycle so that repair of DNA can take place during this day. The next role played by the p53 protein is the activation of the DNA repair enzymes, but if the DNA damage is not able to be repaired, then the third function of CKI inhibitors is activated, and that is the initiation of programmed cell death or apoptosis of the concerned cell. Apoptosis is a strategy which is taken up by the cell in order to prevent the Figure 2.119 diagrammatic representation of the role of p53 in cell cycle inhibition. So we can conclude that p53 ultimately prevents the mutations to be transferred to the daughter cells. We witness the fact that when p53 gene has been mutated and the p53 protein is found to be imperfect or absent, the mutations lead to cancer. At present, it has been established that the p53 gene is a single gene which is chiefly mutated in cancers, and thus it finds the most urgent place among the scientists as a hot topic of research and also in devising new treatment strategies for cancer. Protein dimerization partner E2FDP dimers, which prove to be transcription factors of the E2F family that force the cell to proceed into S phase. So we can say that on binding to E2F production of proteins is stopped. Now when the size of the cell increases, the RB is phosphorylated to inactivate it. On phosphorylation, the complex of RB and E2F breaks, and thus E2F is released, which results in activation of genes for production of certain proteins which will release the block of G1S transition. Role of RB in cell cycle inhibition. Retinoblastoma RB proteins are a group of tumor suppressor proteins just like p53 proteins. RB influences some of the other proteins which allow the cell cycle to proceed further. It was found that the size of the cell is controlled by RB protein. When this RB protein is dephosphorylated, it becomes active and binds to the transcription factors like E2F, which in turn switches on certain genes and thus produces proteins of a specific kind. RB binds and inhibits E2 promoter binding. Figure 2.120 diagrammatic representation of role of RB protein in cell cycle inhibition. Role of TGF-beta polypeptide factor in the cell cycle inhibition. TGF-beta, a polypeptide factor, proves to be an inhibitor of proliferation of the animal cells. It hinders the multiplication of different epithelial cells by checking the progression of cell cycle. In G1 phase, the CDK inhibitor P15 binds to CDK4, 6-cyclin D complex, thus activating the inhibitor polypeptide TGF-beta. Now as they are seized, no RB phosphorylation occurs leading to arrest of the G1 phase. So, in the normal cells TGF-beta via a particular signaling pathway ceases the cell cycle at the G1 stage so that the proliferation of cells is stopped, further inducing differentiation if the cell is found to be okay as regards to its genetic constitution or else encourage apoptosis of the defective cells. Thus, any kind of mutation in TGF-beta results in loss of cell cycle control leading to cancer. Figure 2.121 diagrammatic representation of role of TGF-beta polypeptide factor in the cell cycle inhibition. Role of CHK1 kinase in cell cycle control at G2 checkpoint. If there is any DNA damage detected in G2 phase, the phase is arrested instantly and the arrest continues till all the damage in the DNA is repaired. If this phenomenon in the G2 is not followed, then the cell enters mitosis carrying the flaws in DNA to the next generation cells. This anomaly may lead to chromosomal abnormalities as far as their segregation is concerned, thereby leading to defects in chromosomal segregation and subsequently causing a reduction in the viability of the cell. In 2000, O'Connell et al. Furnished a complicated model for the way many proteins of different checkpoints coordinate to guarantee the activation of CHK1 protein. This protein is an effector kinase for the G2 DNA damage checkpoint and thus initiation of the checkpoint signaling cascade. The molecular mechanism involved in controlling the progression of cell cycle through the G2 checkpoint checks the entry of the cell into mitosis when there is unreplicated or damaged DNA. Fig 2.122. In response to DNA damage or incomplete replication, a protein kinase called CHK1 is activated and thus results in the arrest of the cell cycle at this particular G2 checkpoint. When CDC2 is in the inactivated state, then the succession of the cell cycle to mitosis is blocked and the cell gets arrested in G2 phase. Figure 2.122 diagrammatic representation of the role of CHK1 and CHK2 in cell cycle inhibition. Figure 2.123 diagrammatic representation of role of CHK1 in cell cycle inhibition. Chromosomal organization of DNA chromosome, an overview. If we look upon DNA as the blueprint of life, then chromosomes are the entities that hold the blueprint together. Waldeyer first coined the term chromosome in 1888 when he saw colored bodies stained with an aniline dye under the microscope. Chromosomes are responsible for physically carrying the DNA and all associated proteins. Even the tiniest of beings, bacteria, can hold anywhere between 130 kbp to 14 mbp of DNA while the human genome holds over 3 billion bits per second, which would measure approximately 2 meters if placed in tandem. Fig 2.124 shows the sizes of the chromosomes of various organisms. It is bewildering how this massive amount of DNA is assorted and physically packaged in cells of microscopic sizes and yet made accessible for various life processes like replication, transcription, etc. Even though our understanding of the structure and organization of DNA packaging in chromosomes is constantly evolving, the last few decades have greatly enhanced our knowledge in this field. Figure 2.124 the average size of the genome of organismal groups. The scheme depicts the average size of the genome of various groups of organisms and shows what proportion of the genome codes for peptides. There are many variations in the form of DNA packaging observed in different life forms. The bacterial genome comprises a single DNA molecule which is present in the form of a single circular covalently closed chromosome and compacted with the help of packaging proteins. On the other hand, the haploid human genome has its DNA segregated into 23 units that form morphologically distinct linear chromosomes on compaction. Fig 2.125. Then, there are specialized chromosomes like the polyene chromosomes that perform specialized functions. However, chromosomes universally perform two fundamental functions: the precise transmission of genetic information and accurate control of gene expression. Specialized regions containing repetitive sequences help form structures such as telomeres, replication origin, etc., which help the chromosomes in the execution of their functions. The level of chromosome compaction also varies depending on which areas are needed for active transcription and which areas will remain inactive. Our current ideas of chromosomal structures revolve around understanding how chromatin is arranged in a structural domain in the three-dimensional nuclear spaces and how this structural landscaping controls different facets of gene expression. Figure 2.125 varying forms of chromosomes. A. The diagram is a schematic representation of a bacterial nucleoid. It depicts a bottle brush model of the nucleoid with supercoiled loops that are interwound and radiate out of a dense core. B. A schematic sky image of a normal female karyotype. DNA supercoiling. Now that we appreciate the gigantic size of the genetic material that must be packaged into a comparatively minuscule cellular space, it stands to reason that the DNA strings must be highly coiled and condensed. Yet, the coiling should still be functionally important domains of the DNA accessible to proteins. This feat is achieved through the process of supercoiling, which implies the coiling of a coil. To understand the process, let us visualize a double helical DNA. An axis passes through the middle of the helix. When this axis is folded upon itself, it results in a supercoiled DNA. Fig 2.126. This can be explained by using a small linear double helix with 2 to 3 turns. If both ends of the helix are twisted in the direction of the helix winding, the number of turns in the helix will seem to increase. This supercoiled helix is overtwisted and under strain. If, however, the helix is twisted in the direction opposite to its coiling, it will appear to unwind. This state of the coil with lesser number of turns is a relaxed state. If the molecule is consistently overtwisted, it will relieve the molecular strain by twisting upon its own helical axis, thus creating a positive supercoil. Likewise, an undertwisted moiety will result in a negative supercoil. Most basic processes like replication and transcription require the unwinding of the double helix. This in turn results in overtwisting of the domains lying ahead. Thus, supercoiling is an integral aspect of the tertiary structure of the DNA that is omnipotent in cellular DNA and tightly regulated by the cell. Figure 2.126 the supercoiling of DNA. A linear double helix is shown on the left-hand side with an imaginary axis passing through it. The right-hand figure depicts a supercoiled DNA where the axis has folded on itself. To understand the physiological relevance of supercoiling, let us focus on closed circular B form of DNA which has 10.5 bits per second per turn. Fig 2.127. The underwinding of this DNA at any point will result in a strain. For example, if the DNA has 84 bits per second, in a relaxed state it will consist of 8 helical turns of 10.5 bits per second each. If the DNA is now underwound and one of these 8 turns is removed, then the 84 bits per second will be divided into 7 helical turns containing 12 bits per second per turn instead of 10.5 bits per second. This alteration will result in a thermodynamically strained structure. This strain can be compensated in 2 ways. Either the strands can simply separate over a stretch to resort to 10.5 bits per second per turn, or the axis of the double helix can coil on itself to realign the base stacking to approximately 10.5 bits per second per turn pattern. Figure 2.127 DNA underwinding. A relaxed 84 bps DNA segment with 8 helical turns. B. The underwinding of the DNA by removing one turn results in strained DNA which is compensated by C. Separation of the strand over 10.5 bps or D by forming a supercoil. DNA supercoiling can be better understood with the help of a branch of mathematics called topology. Of special interest is the concept of linking number LK, which denotes the number of helical turns in a closed circular DNA when no supercoiling is present. Fig 2.128. This property does not vary even if the DNA is twisted or deformed as long as the 2 strands of DNA remain intact. By standards, if the DNA strands are twisted in a right-handed helix, that denotes the coiling of the helix axis while twisty finds the local twisting or the spatial arrangement of the neighboring base pairs. When supercoiling happens, the linking number of the DNA changes causing a strain which is compensated by variation in supercoiling right or by a change in the twist patterns. This gives rise to the equation LK equals TW plus WR. In all systems, the supercoiling is regulated by a class of enzymes called topoisomerases which play an important role in the processes of replication, DNA packaging, etc. The genre of topoisomerases is divided into 2 classes, type 1 and type 2. Type 1 topoisomerases act by creating a nick on one strand, twisting it around the uncut strand, and rejoining it, thus changing the LK by 1. Type 2 topoisomerase acts by cutting both strands of the DNA, twisting them around one another, and rejoining them, thus changing the LK by 2. Linking number is a positive integer. However, if the strands are twisted in a left-handed helix, as in zDNA, the linking number is a negative integer. Figure 2.128 depiction of linking number LK. Of the molecules shown, physically interact at one junction and have an LK at 1. B. If one strand of DNA is kept unwound, then the number of times the second strand passes, it defines the linking number for that molecule. 2 structural components make up the linking number, twist TW and ride WR. Ride. Figure 2.129 the mechanism of strand passage through topoisomerase core. The type 2 topoisomerase blue and yellow core captures G segment gate DNA green. It then sequesters the transfer of T segment indicated in shades of pink to denote the movement of DNA. Then they bind 2 ATP molecules, thereby shutting the N-terminal gate yellow, which is followed by the double strand cleavage of the gate strand. The T segment passes across the cleaved DNA and, together with the ATP hydrolysis products, is then released. Of the coiled structures is maintained. Most of our knowledge of the organization of prokaryotic chromosomes comes from the studies conducted on E. coli. The single circular chromosome is packaged in the form of a nucleoid which is defined as the area of the cell where the chromosome DNA is located. Organization of the prokaryotic chromosome. Having understood the basic dynamics of DNA supercoiling, we move on to the question of how the DNA is packaged in prokaryotes lacking a defined nucleus and how the stability. Figure 2.130 packaging of the bacterial chromosome. A circular chromosome without any compaction. B. The DNA associated proteins form a central scaffold and the bacterial DNA loops around them. Seed the loops are further supercoiled to form a condensed structure. The packaging of the bacterial genome into the small nucleoid space occurs via two processes. The first is the process of supercoiling discussed in section B2, and the other is the interaction of the DNA with packaging proteins. The main histone-like packaging proteins present in E. coli are WHO, IHF, integration host factor, FIS factor for inversion stimulation, and HNS. These positively charged proteins interact with the negatively charged DNA and together with the topoisomerase and gyrase enzymes maintain the supercoiling homeostasis of the bacterial genome. For example, the WHO protein works with topoisomerase I and introduces acute bends in the chromosome which generate the tension required for the negative supercoiling. During normal growth, most of the bacterial genome is negatively supercoiled. The negative supercoiling of DNA gives rise to plectinemic loops forming a network of supercoiled domains that are topologically insulated from each other. The current view is that these interwound loops are not rigid. They keep changing depending on the genetic transaction that occurs within and between them. Figure 2.131 proteins associated with bacterial chromosomes. Several proteins like U and HNS are associated with the bacterial DNA to maintain its topological structure. Hierarchical packaging of the eukaryotic chromosome. When DNA from eukaryotic cells is isolated using isotonic buffers like 0.15 mkcl, it is found to be associated with nearly equivalent proportions of protein in an extremely compact complex called chromatin. The DNA in eukaryotes exists in the chromatin state throughout the interphase of the cell cycle. However, for all the tangled DNA to accurately partition into two cells during mitosis, the DNA needs to condense into ordered structures called chromosomes during the prophase of the cell cycle. As discussed, the total length of DNA present in a diploid human cell is about 2 meters. This is divided into 23 pairs of chromosomes, each containing anywhere between 15 to 85 millimeters of DNA. The largest human chromosome, of approximately 85 millimeters long DNA, is packaged into a distinct meiotic chromosome, 10 millimeters long and about 0.5 millimeters in diameter. Even so, the chromosome has specialized structures and its dynamic topology allows access to proteins required for life functions. This is achieved by a hierarchical and orchestrated process of packaging with the help of different types of proteins. The nucleosome assembly. The most predominant proteins found attached to the eukaryotic DNA are the histone proteins. They represent a family of basic proteins, rich in positively charged amino acids such as lysine and arginine. This positive charge helps the protein interact with the negatively charged backbone of DNA. There are 5 principal types of histones: H1, H2A, H2B, H3, and H4. The histones form a highly structured assembly and the DNA loops around it to give rise to the beads on string moieties known as the nucleosome assembly. The nucleosome consists of a string of DNA wrapped around a protein core like a thread on a spool. The disc-shaped core is an octamer comprising 2 copies of H2A, H2B, H3, and H4 each. Histone protein subassemblies come together to form the histone core with the H3 and H4 forming a tetrameric subassembly and the H2A, H2B dimeric subassembly joining it to complete the histone octamer core. The DNA wraps around this core approximately 1.65 times using around 146 bits per second length. The H3 and H4 tetramer interact with the middle and the rear ends while the rest of the DNA is bound to the H2A, H2B dimer via hydrogen bonds. Consecutive nucleosome cores are connected by short segments of linker DNA which harbors the linker histone, H1. Figure 2.132 the hierarchical packaging of the eukaryotic chromosome. Assemble these DNA wounds around the nucleosome complex giving rise to a beads on string structure. This nucleosome forms a solenoid complex of 30 newton meters diameter. The 30 newton meters fiber further forms 300 newton meters long loops around the non-histone proteins undergoing several levels of compaction to form the chromosome. Figure 2.133 the nucleosome assembly. The nucleosome consists of an octameric core comprised of the H2A, H2B, H3, and H4 units with about 146 pp's of DNA wound around it. Each histone protein has an N-terminal tail that provides a guide for the DNA strand to wrap around the core by creating grooves similar to those on a screw such that the DNA wraps in a specific pattern. Thus, the beads form ellipsoidal bodies measuring 110 A in diameter and 60 A in height. Figure 2.134 crystal structure of the nucleosome assemble. The image shows a schematic representation of the X-ray crystal structure of the nucleosome core. All reference frames are in alignment and computed by the PCA of the globular core of the histone octamer. Packaged into a spiral arrangement having 6 nucleosomes every turn. The nucleosomes fold on the inside with the help of a linker, H1 histone giving rise to a 30 nanometers chromatin, Fig 2.135. A unit nucleosome and a unit of the H1 together are called the chromatosomes. The solenoid structure. The electron microscopy data show that the chromatin fibers have a diameter of approximately 30 nanometers. The 10 nanometers nucleosome fiber is wrapped into a highly structured solenoid or a supercoil. The solenoid model depicts the nucleosome as Figure 2.135 the solenoid model. The nucleosomal core arranges in the form of a solenoid by bending over the H1 linker histone, thus giving rise to the 30 newton meters fiber. The chromosomal structure. The 30 nanometer solenoid chromatin again undergoes several orders of supercoiling to finally form the metaphase chromosome. The chromosome at the metaphase stage has the highest order of condensation. This is composed of loops of the 30 nanometers chromatin solenoid radiating from a central scaffold. The central scaffold contains non-histone proteins which anchor the chromatin at specific AT-rich regions called the matrix-associated regions (MAR) or scaffold-associated regions (SAR). Each loop emanating from the scaffold structure is about 100 to 300 nm, approximately 60 kilobits per second long. It has been recently shown that this model of the chromosome is neither a rigid model nor a random one. The chromosomes organize themselves in the three-dimensional nuclear space in the form of distinct territories with defined interactions with the nuclear subdomains. This gives rise to what is known as topologically associated domains (TADs). TADs are essentially self-interacting regions of DNA which are far apart from each other in terms of their two-dimensional length but are brought together by the process of looping and three-dimensional organization of the chromosome. This helps to form two compartments, an open or transcriptionally active compartment and a closed or transcriptionally inactive compartment. So, TADs can be simply visualized as long stretches of DNA that come together in a common 3D space so that the genes that they house can be triggered or silenced in sync. The heterochromatin and euchromatin as discussed earlier, one of the most important tasks of the chromatin and chromosomal structure is to allow the accurate control of gene expression. Therefore, the chromatin fiber must make allowances for the proteins to reach and transcribe a segment of DNA required to be active while leaving other non-relevant segments inactive. In most regions, the chromatin appears to be less compactly packaged and relatively more dispersed in the nucleus. These regions are transcriptionally more active and are known as the euchromatin. On the other hand, the more densely packaged regions of chromatin resembling the level of condensation seen in the chromosomes are transcriptionally inactive and known as the heterochromatin. These alternate states of packaging are achieved by various mechanisms including histone modification. Heterochromatin can be formed with the help of increased methylation of one histone, decreased acetylation of histones, and hypermethylation of the cytidine bases of the DNA. Chromosomal banding. The study of the structure of chromosomes requires rigorous techniques to stain and visualize them under a microscope. This is done by enzymatic digestion or denaturation of the chromosome followed by staining with DNA binding dyes. Because of the variations in the extent of packaging, this procedure produces light and dark banding patterns on the chromosomes. Each chromosome has a unique banding pattern which helps in its identification. Following are some of the banding techniques used routinely for the study of chromosomes. G-banding. Controlled Trypsin digestion of the chromosomes is performed, followed by staining with Ginza. The bands that take the dark stain are called G-bands while the lightly stained bands are called G-negative. G-bands are mostly AT-rich segments of DNA. Figure 2.136 G-banding pattern of the human chromosome is observed under a microscope. Ginza is a protein stain that darkly stains the heterochromatic or transcriptionally inactive regions and lightly stains the euchromatic or transcriptionally active regions. Can be achieved by using GC-specific binding dyes. T-banding. This banding technique is a more severe form of R-banding used to identify the telomeric regions of DNA. This is achieved by either a more severe heat treatment of DNA prior to Ginza staining or using a combination of standard and fluorescent dyes. C-banding. This technique is used to stain constitutive heterochromatin of the centromere. The DNA is denatured with a barium hydroxide solution before being stained with Ginza. Q-banding. This technique requires the staining of DNA with fluorescent dyes such as quinaprine, dapple 4-feet, 6-diamino-2-phenylenol, or Hoechst. T-banding. This banding technique is a more severe form of R-banding used to identify the telomeric regions of DNA. This is achieved by either a more severe heat treatment of DNA prior to Ginza staining or using a combination of standard and fluorescent dyes. C-banding. This technique is used to stain constitutive. The heterochromatin and euchromatin as discussed earlier, one of the most important tasks of the chromatin and chromosomal structure is to allow the accurate control of gene expression. Therefore, the chromatin fiber must make allowances for the proteins to reach and transcribe a segment of DNA required to be active while leaving other non-relevant segments inactive. In most regions, the chromatin appears to be less compactly packaged and relatively more dispersed in the nucleus. These regions are transcriptionally more active and are known as the euchromatin. On the other hand, the more densely packaged regions of chromatin resembling the level of condensation seen in the chromosomes are transcriptionally inactive and known as the heterochromatin. These alternate states of packaging are achieved by various mechanisms including histone modification. Heterochromatin can be formed with the help of increased methylation of one histone, decreased acetylation of histones, and hypermethylation of acidity bases of the DNA. Chromosomal banding. The study of the structure of chromosomes requires rigorous techniques to stain and visualize them under a microscope. This is done by enzymatic digestion or denaturation of the chromosome followed by staining with DNA binding dyes. Because of the variations in the extent of packaging, this procedure produces light and dark banding patterns on the chromosomes.